Then silver nanoparticle is, and the inhibition zone I obtained is 2.6. Then negative control ke liye I have taken silver nitrate, and the inhibition zone I got is 1.5 centimeter. In case of Staphylococcus aureus, which is a gram-positive bacteria, again I have taken the positive control ampicillin, and inhibition zone I got in this case is 3.0. Then silver nanoparticle, again I have got 2.5, and negative control is silver nitrate. In this I got 1.6 centimeter inhibition zone. From this I have this result I got key silver nanoparticle was successfully synthesized from chlorella species, and it shows the antimicrobial activity against E. coli and Staphylococcus aureus. Thank you.
Thank you, Pallavi. You switched on your video again. You can unshare your... If you need to see, sir, let it be.
Yeah.
Let it be, wait. Shared, unshared. Okay, fine. So, Ms. Pallavi, just one question. Which strain of E. coli you have used?
Ma'am, this is a standard one.
In type.
Can you name that standard strain?
Recently, I am not doing, I have taken the standard, but after some time, since I got the result, now I will go to CDRI for further analysis where I will take the MDR strain.
Okay, can you tell me how much is the yield of your nanoparticle?
How much?
Yield of your nanoparticle.
Yield of nanoparticle. So 100 100 milligrams into 100 or I can say 150 ml of 150 15 लीटर ऑफ एक्वा जो एल्गल एक्सट्रैक्ट है जो सैंपल लिया था उससे मुझे ले दे के मतलब 100 मिलीग्राम नैनो पार्टिकल मिल पाता है. बहुत ही जो जितना हम लोग लेते हैं कलेक्ट करते हैं सैंपल उससे बहुत कम उसमें बायोमास मिल पाता है, फिर उससे भी बहुत ज्यादा कम silver nanoparticle मिल पाता है. Yield बहुत कम.
I understand, ma'am. Whenever you are doing for the large scale production, then the cost of the nanoparticle will be higher. If you don't know the yield, exact yield means if yield is higher means the cost of production of nanoparticle is lower. So in that way, your method can be used for the further scale up of the process. So this is all from us.
Thank you, sir.
From where you have done the SEM and what was the analysis? Just repeat that.
I have for the SEM analysis, I went to BBAU University, Lucknow.
From there you have done?
Yes.
Okay, fine. Thank
you.
You can leave.
Who is next?
What is the OP number? 22.
So, Astha Jaiswal, Miss Astha Jaiswal, can you start your presentation?
Hello, yeah, I'll just share my presentation. Okay, is it visible and am I audible?
You are.
Okay. So, good morning, everyone. I'm Astha Jaiswal, fourth-year student BSc. I work on induced mutagenesis for breeding of leguminous crops towards circular bioeconomy and sustainability. And this work has been done under the supervision of Professor Vijayarani Rajpal ma'am. Is it not? Yeah. So, as we all know, that hunger and malnutrition are two of the major global concerns, which are also listed under the Sustainable Developmental Goal 2, that is zero hunger. And in Asian and African subcontinent, cereals and legumes form the staple diet of the vegetarian population, with lentils, soybean, and chickpea serving as the major protein sources. Lentils are an excellent source of protein as they contain approximately 24 to 26% protein and are also rich in essential amino acids, fibers, and minerals with excellent digestibility. And it is a key Rabi pulse crop that is also very important in India's food for ensuring India's food and national security. And the key lentil producing states in India include Madhya Pradesh, Uttar Pradesh, Bihar, Jharkhand, and West Bengal. And as you can see from the graph in the chart, the national productivity of lentil has increased from 12 to 18 lakh tons over the last 10 years. However, the Indian average productivity is still lower than the global average productivity, leaving a productivity gap of around 200 kilograms per hectare. This productivity gap highlights the need for crop improvement interventions in lentils. However, due to prolonged domestication and selective breeding, the genetic base of lentil have eventually become narrow. So as to broaden the narrowed genetic base of lentil, and many breeding techniques have been evolved. However, the conventional breeding technologies are not sufficient to introduce novel genetic variabilities quickly. So here comes an age-old technique of induced mutagenesis that offers a promising approach to introduce novel genetic variability in a short period of time. That is also quite inexpensive process. So the objectives of my study is to conduct a comprehensive literature review and to evaluate the effect of different doses of gamma rays on chromosomal behavior in lentil root tip cells, to assess the frequency and spectrum of chromosomal aberrations observed, and to analyze their dose-dependent abnormalities, and finally to identify the optimal gamma radiation dose that induces significant chromosomal variation while maintaining cytological stability. So for methodology, my work is an experimental work, and the plant material used here is Lens culinaris variety PDL1, the seeds of which were procured from the Division of Genetics, Indian Agriculture Research Institute, and further followed by gamma irradiation of varying doses. Then germination on moist filter paper on Petri plates, followed by root tip excision for mitotic analysis. And these are my results. So here, as you can see, a wide spectrum of mitotic aberrations were observed, and their frequency increased with progressively by increasing the radiation doses. And abnormalities observed included C mitosis that indicates severe spindle disruption. Similarly, diagonal anaphase and Legards also confirms the spindle malfunction and improper segregation that shows the angiogenic effect of gamma rays. Apart from that, chromosomal bridges were also observed at both anaphase and telophase stages. That bridges are formed due to misrepair of the double-stranded breaks caused by the radiation, and that leads to dicentric chromosome formation. And chromosomal breaks again reflects the clastogenic effect of the radiation. Apart from this, micronuclei was also observed, and micronuclei arises from lagging chromosome or acentric fragments that are unable to get incorporated into the daughter nuclei. And at higher doses of radiation, severe cytotoxic effects were observed on the chromosomes as abnormalities such as Carey-Rexes and vacuolated nuclei that indicates extensive chromatin disintegration and cytotoxic stress. So overall, these findings demonstrate that gamma rays induce various clastogenic and angiogenic effect on the chromosome, and in a dose-dependent manner, leading to chromosomal instability. While higher doses causes while higher doses causes genomic damage, controlled doses or moderate doses such as 200 to 300 grays can generate beneficial mutations and control genetic variability that can be exploited in the future mutation breeding programs. So I would like to thank my supervisor, Professor Vijayarani Rajpal ma'am. I'm also thankful to the Department of Botany, Hansraj College, for providing me with the necessary facilities. I also thank Dr. Dhamendra Singh, sir, Division of Genetics IRI, for providing me with the seed material. And I also thank Dr. Bhupendra Singh, sir, Nuclear Research Laboratory IRI, for facilitating the gamma irradiation process. I also thank the organizers of this conference for letting me present my work over here. Thank you.
Thank you, Ms. Asta. So you just tell me, you have shown me 5-6 objectives in your first slide, right? So you just don't show me, you just tell me the overall work you have concluded, the overall, all the objective you have shown us.
Literature review and after that, I dosage range of gamma irradiation, the crop, the leguminous crop that I need to work on, as there is very less study have been done on this particular crop, and
you have not done yet.
No, I did that. Then only I.
That's what I'm saying. All the 5 objectives you have completed.
Yeah.
Have shown us all the 5 objectives result.
Yeah.
Can you show me the slide of your objective, please?
Yeah, sure.
Yeah. So finally, you're working for the circular bioeconomy principles, right? So how does your dose, what was the dose which you have fixed, and how you have observed this? You can tell me in a single line. Can you hear me, Asta? She's not able to hear.
My question is that how your study is related with the circular bioeconomy. Can you comment on this?
Hello. I'm really sorry. I got disconnected. Hello. Am I audible now?
Yes, you are audible. I have a last question from you. How your
Yeah.
study is correlated with the circular bioeconomy? Can you comment on it?
So basically, mutation breeding would help to enhance the genetic variability so that better varieties can come that would have enhanced productivity and that would lead to betterment of the economy for the country, for the production point of view of the food and legumes.
Okay, you can leave talk and
Okay,
there is
thank you so much,
next.
sir.
Who is next?
Is there? Priyagri? OP20. 20 is not there? Just check so that we'll mark her absent then.
So OP 23rd, T. Angeline Sweta, you can start your presentation.
Okay, sir.
Yeah, Shweta, you can start. Share your slides.
Okay, ma'am. I'm audible. Okay. Good afternoon, everyone. I'm Engineer Shweta from Department of Microbiology, Alagappa University. I am working under the guidance of Dr. A. Arun. My research topic is on sustainable lactic acid and polylactic acid production by lactic acid bacteria supplement. Ma'am, can you repeat again?
Yes.
Hello?
Yes, you can proceed.
You can proceed. We are able to see. Yeah.
Okay. Supplemented with mixed renewable substrate, mixed fruit waste, optimization, downstream process. Okay, ma'am. Okay, ma'am. Downstream processing, conversion, and characterization. Generally, plastics are categorized into two types, petroleum-based and bio-based. Petroleum-based plastics are produced using fossil fuels. It uses 98% of fossil fuels, which is to depletion of natural fossil fuels. And in globally in a year, 20 million tons of petrochemical plastics are produced, and which is being dumped which leads to enormous amount of plastic waste. In a year, 8 million tons of petrochemical plastics are dumped into the oceans. If it continues by the year of 2015, there will be a result of endangered species in the. Okay. Okay.
Hello, Shweta.
Shall I?
Switch on your video as well. And your slides are not moving. It is in first slide itself. Switch on your video.
Hello, ma'am.
Yes, Shweta, just share it. Your slide was not moving. It was in first slide itself. And you should not switch off your video, right? It should be
No,
on.
ma'am. No, ma'am. It is on only. Now,
You share your slides.
is my shares are visible, ma'am?
Not yet. Yeah, it is. Now it is. Make it slideshow and just check whether it is moving or not. Yeah, make it slideshow mode.
Okay, ma'am. Okay. Is it visible, ma'am? It's moving.
It is visible. Go to the first slide. It is in second slide. This one is second. Okay.
Okay.
Yeah, now it's fine.
Yes, ma'am.
Carry on.
Shall I start, ma'am?
Yeah, yes, start.
Okay. So these problem challenges mankind to produce bioplastic from renewable resources. These bioplastics can be produced from renewable resources like agricultural waste, lignocellulosic waste, aquaculture waste, and other bio wastes like food waste, municipal waste, etc. And the carbon needed for the production of bioplastics is produced through photosynthesis, and when they degrade, it releases less amount of carbon dioxide. When compared to petrochemical plastic, it has less carbon footprints, and which helps in climatic changes reduction means prevention in climatic changes in the environment. When compared to other bioplastics, polylactic acid production used to increase annually. Polylactic acid is one of the biodegradable plastic with aliphatic polyester group, and it is produced because of its best bioabsorbable property, which is from the monomer called lactic acid. Generally, lactic acid is available in two forms, L and D forms. This lactic acid is otherwise known as 2-hydroxypropionic acid. It can be produced by chemical and biological method. The disadvantage in chemical method is because it produces race mixture of lactic acid, where in biological method it produces optically pure lactic acid by using certain microorganisms. Methodology. In our study, we have collected mixed fruit waste from the fruit juice shops, and the physiochemical parameters has been analyzed. After analyzing, the mixed fruit waste was enriched using MRS broth in agitate condition at 37 degrees for 24 hours. After enrichment, the broth was serially diluted and spread plated on MRS agar media with bromoclosal purple as an indicator. After isolation, all the isolates were subjected to titrable acidity in order to find the potential lactic acid producer. The best producer has been further identified using 16S rRNA sequencing and further used for our optimization study. After optimization, the best optimum condition has been identified. From there, the lactic acid has been extracted and purified using phase partitioning method. After extraction and purification, the purified lactic acid is being converted into polylactic acid using melt polycondensation method. Both the monomer and the polymer produced has been undergone many characterization studies. These are my results in the physiochemical parameters of mixed fruit waste after the collection of from the area, and totally 5 isolates has been isolated from in our study, and they are assessed for their titrable acidity, from which we have identified FWLP4 has produced highest amount of lactic acid of 23.6 gram per lactic acid per liter in 48 hours of incubation. This is our optimization results. In our optimization, we have used mixed fruit waste as a sole substrate, where substrate concentration has been optimized from 10 to 100 degrees Celsius, different pH where we have optimized, and different incubation periods were optimized, and in both agitate and static conditions were done. From this, we have identified the best optimum condition in is
You need to be fast.
48 hours of. Okay, ma'am.
Shweta, you need to
at
make it
48
fast.
hours. Okay.
Yeah, carry on.
Okay, 48 hours with the production rate of 26.3 gram per liter of lactic acid. After the production, from the best to optimum condition, the purified LA has been extracted and purified, and with the FTR, it has been FTR and NMR, it has been conformed with peaks at carboxylic peaks at 1732 centimeter inverse, which has been
Shweta, your voice is not at all clear.
Standard and in I can identify using their methyl and methyl. Now it is clear.
Yeah, you conclude your slide first.
Okay, ma'am. Various then NMR study has been done used for the produced PLA and LA, and DSC and TGA has been done where it has observed that it has same rate of heat degradation, and their class transition, crystallization temperature, and melting temperature has been observed with the is compared with the standard PLA. PLA is a biodegradable thermoplastic polymer with aliphatic polyester group, and it can be produced from renewable resources. In our study, we have used mixed fruit waste as a substrate in order to produce lactic acid, and in order
Wind
to
up now. Wind up. Your time is up.
Okay, ma'am.
Yeah. So, Asta, you have in conclusion also you have said that mixed fruit waste is there.
Yes, ma'am.
So from where you have collected this fruit waste? Can you tell me?
Ma'am, fruit juice shop we have collected, ma'am.
In question is the fruit juices, juice shop, wherever you have collected, maybe the fruit may be varying. So how can you optimize or make sure that this fruit waste will be getting everywhere? Are you getting my question?
Yes, ma'am. Right. And we have collected all types of fruits, which were means all seasonal type of fruits in every season we have collected, and we have blended and we have optimized in our study, ma'am.
Typically, what fruits are there?
Like apple, mango, grapes, pomegranate, fig, dates, then like custard apple, watermelon, pineapple, orange.
Custard apple is not juice shop.
Yes, ma'am.
Acha. They prepare juice from there? Custard?
Yes, ma'am. Yes, ma'am. In our side, they used to prepare.
Is it?
Yes, ma'am.
New thing for me. Okay, fine. So what is the efficacy which you got? The conventional plastic on environment, how you think that whatever you have found out, right, that is environment friendly? How will you prove that answer?
Ma'am, we are now after production, now we are doing degradation study from our produced PLA, and we are studying the rate of degradation with from which we can conclude it is completely biodegradable. So, and it will help to save the environment from conventional plastics, ma'am.
Hello, can you hear me?
Yes, ma'am, I can hear you.
Yes, very nice presentation actually you gave here. So I have this one question from this conclusion that I can see here. Is this you written here the status octanoate was utilized as a catalyst in melt poly condensation polymerization. Is there any specific enzyme is there so that poly condensation polymerization can takes place, so that's at reduced pressure that you can produce the PLA?
Ma'am, means enzyme you are asking, ma'am.
Yes, yes. Is there specific enzyme that you know you've gone through this, you review, but you
Yes,
studied
ma'am.
the literature?
Yes, ma'am.
Yes,
Enzymes were there, they have used
lactate.
lactase. They have used, ma'am.
Okay. All right, fine.
Okay.
Okay, you may leave.
Thank you, ma'am.
Megha. Megha
Yes,
Arora, are you there?
yes, ma'am. Yes, ma'am.
Yeah, just switch on your video.
Ma'am, just 1 minute. Ma'am, my screen is visible.
Oh, you are also not visible. Your screen is also not visible.
Oh. Minute. Just 1 minute. मैम आई थिंक मैं कुछ शेयर ही नहीं कर पा रही हूँ विंडो कुछ प्रॉब्लम आ रही है
Acha, we are having your presentation. We can share, but you need to switch on your video first, Megha.
ओके मैम जस्ट 1 minute.
We'll share from your side. Huh? Ah, it's done. Now we can see your slide also.
ओके मैम
Slideshow mode.
जस्ट 1 minute.
Yeah. Okay, no problem.
अभी मैम विजिबल है?
It is. It is visible, but make it in slideshow mode. ये आधा क्यों दिख रहा है? It is half away. I can see. Yeah, now it is fine. You switch on your video. Your video is off again. We should be seeing you while you speak.
मैम हाँ, आई थिंक कोई ऑप्शन ही नहीं आ रहा है यहाँ पे फर्दर। वो अगर मैं उसको
Can you see your slide? Slide तो ठीक दिख रहे हैं. नहीं हाँ.
मैम स्लाइड्स दिख रहे हैं, बट अगर मैं स्लाइड्स को वो कर रही हूँ तो फिर
चलो
मेरी
कोई बात नहीं.
स्क्रीन।
We'll see. आप कंटिन्यू करो.
मैम मैं स्टार्ट करूँ?
Yes.
Okay. Good morning to all of you. I am Megha, research scholar in Life Science Department in JC Bose University of Science and Technology. And today I am presenting my oral presentation, Bibliometric Perspectives on the Microalgae based on the phytotoxicity assessment, trends, research, hotspots, and the future directions. Why these studies are necessary? Basically, mostly जो textile dye है वो परसिस्ट करती है, लार्ज इफ्लेंट्स में रिलीज हो रही है, और उस डाई को फर्दर मतलब उसको फर्दर यूटिलाइज, उसको ना तो उसका प्रॉपर उनको फर्दर यूटिलाइज किया जा रहा है, ना उनको फर्दर उनका रिमूवल
Are
किया
you in Hindi medium university? Is it?
नो मैम, बट आई प्रेफरली टू कन्वे इन प्रॉपर
In Hindi? We are going throughout English only.
Megha, listen.
ओके
Megha, can
ओके
you
ओके ओके मैम
try to be finish in English, right?
ओके मैम ठीक है। Textile dye are persisting a xenobiotic pollutants with azodye. Azodye dominant in the industrial discharge. Although many studies have been report the effective color removal, most of the focus on the decolorization efficiencies, but decolorization is not equal to detoxifications. So very limited studies validate the ecological safety, environmental safety through the phytotoxicity. So these structured bibliometric analysis required the global research trend, identify the critical research gap, why the decolorization, why only focus on the decolorization study, not on the detoxification, and which parameters affects the environment, and what is the safety precautions used to release large effluents of dye. Next, the study objective of this bibliometric analysis. First is called the quantify the global publications growth, key which sectors contains the high dye large effluent, mainly just say textiles dye. Identify the thematic evolutions, key what is the origin, map research hotspots, which regions contain large effluents. Analyze the geographical journal distributions like SCI, elsewhere journals which publish more papers on the dye decolorization, detect the research gap and the limitations. And these are the research evolutions. First, we study the literature's search paper, then further to collect the database on Scopus and study the few keywords like strategies like microalgae, dye decolorization, detoxification, then data extraction, and further to analyze the trend, hotspots, and to identify the gap. And second one is called the ecological risk, like mainly to studies was focused on the decolorization, but few research gaps are present on the mechanism which use in the biodegradations, and why these degradation products are harmful to the other like parameters like human health and other ecological factors, and further to validate these phytotoxicity validation, and further to analyze the ecological risk framework. So the shifting of dye removal efficiency into the ecological risk validations. These are the some thematic clusters. Like first is called the bioremediation. Matlab first we search the paper according to the bioremediation, which contain bioabsorption study. The algal surface absorb the dye on the cell surface. further textile wastewater, and then dye reduction. These few cluster means these few keywords which was used to study and to search the paper. Then cluster 2 is called the algal bioremediation, like different algae use, different algal work used for the dye decoloration, phytotoxic remediation, then lipid accumulation, which dye stress affect the lipid accumulation पे, and further the result output after the dye degradation and decoloration, the biodiesel parameters are okay or not, which affect. And then cluster third is called the toxicity assessment, which is the main ultimate parameters. This called the phytotoxicity key. After the removal of dye, these water further are useful for other agriculture sectors, like to check on the seed germination index, root elongation, shoot elongation, and germination percentage. These are the global contribution pattern. Like most of the country contain China, India, Brazil, but India is also large effect on the India is also contain the large number of dyes are released from other
Wind up first, time is up. 2 minutes are remaining. Just complete.
Okay, ma'am. These are the why these studies are important for, like limited metabolic toxicity profiling, few long term ecotoxicity studies, and rare integration with the lipid and biofuel systems, minimum life cycles. That's why we choose these studies. And these are some few research framework was used for, if use these parameters to further phytotoxicity assessment can be enhanced, like LCMC pathway confirmation, integrated phytoremediations, and the future focus shift from decolorization to the ecological risk. Thank you, ma'am.
Yeah, thank you, Megha. Megha, you have just gone for the literature survey, right? What I understand from your work.
Basically, ma'am, bibliometrics means literature survey to analyze and further to.
That I got. So you just tell me which software or what tool you have used for identifying these things.
हम SPSS यूज किया गया है और वेब ऑफ साइंस.
Unshare your slide, please. We would be able to see you. Unshare your slides.
एक मिनट.
Switch on your video. Yes, now we can see you. Yes, Megha. Tell me how you have identified those things. From which tool?
Ma'am, SPSS.
SPSS for what?
मैम मतलब कि पेपर्स, रिसर्च कैप, कीवर्ड्स को आइडेंटिफाई और डेटा को एनालाइज करने के लिए पूरा प्रॉपर.
इसमें क्या डेटा डाला आपने?
बेसिकली मैम पहले तो कीवर्ड्स को आइडेंटिफाई किया जाता है कि कीवर्ड्स को कैसे, कि कौन से, जैसे जो कि हमने क्लस्टर्स यूज करे हैं, जैसे क्लस्टर 1, क्लस्टर 2, क्लस्टर 3, लाइक फाइटोटॉक्सिटी, माइक्रोएल्गी बायोरेमेडिएशंस, एंड क्या नाम है, इकोलॉजिकल पैरामीटर्स लाइक जर्मिनेशन परसेंटेज.
तो वो दिखाइए ना आप एनालिसिस का, वो कहाँ है? जो भी आपने दिया है वो तो सिर्फ स्लाइड में लिख के दे दिया है. व्हाट यू हैव ऑप्टेन, हाउ विल यू कम टू नो?
मैम वो अभी आई थिंक मैम वो पेपर के लिए थोड़ा सा डेटा भी वो कम्युनिकेशन में गया हुआ है, तो इसलिए मैंने उसको ऐड ऑन नहीं किया.
दे सकते थे, ऐड करने की जरूरत नहीं है, बट फ्रॉम वेयर यू हैव सीन दैट वी कुड हैव अंडरस्टैंड ना कि आपने किया है ना. स्क्रीनशॉट भी आप शेयर कर सकते थे उसका. what you have identified. हम exact पूरा result तो नहीं मांग रहे हैं आपसे. Oh, anyway, it should be there. Means whatever you are giving, I'm not getting actually what you want to convey, the actual part, right? So from three different keywords you have mentioned: bioremediation. The second one was, tell me,
Phytotoxicity.
Phytotoxicity, and the third one is
Algal bioremediation, phytotoxicity, or first one is called the parameters.
yeah. So first one is called as parameters.
मैम जस्ट मैं फिर से स्टार्ट शो करूं.
Show कर दे Dr. Kirti, you ask.
Ma'am, first one is called the bioremediation. Second one is called algal biotechnology, means biofuels, and third one is called the toxicity.
Okay, got it. Megha, actually, I have only one questions to you. Thus, you mentioned in the slides, second slide might be written there mechanistic biodegradation that you mentioned. Mechanistic.
Yes, ma'am.
What
Yes,
do you
ma'am.
understand by the mechanistic biodegradation, and how this, define it in terms of any kind of, cellular, in terms of cellular concept, which type of a changes that you observed, and you are actually collecting the previous analogs to define this mechanistic, because mechanistic is important to understand so that you can versed and well go for the phytoremediation and the phytotoxicity study. This is most understood.
Ma'am,
Yes.
ma'am, mechanistic means in algae under the stress condition like dye stress,
Yeah.
other type of stress. So these stress conditions in neutral lipids. Sometimes microalgae absorb the dye and further in algal cells release large amount of azoreductase. These azoreductase are break down the dye and further convert into the some products like aniline, further other types of product. So these products are further can be utilized by these further products can be utilized for the other process like अगर हम consortia grow करें. So
yes, okay. And second is this in continuation of this answer that you gave for this. This is called lipid peroxidation.
Yes,
Lipid
ma'am.
peroxidation. Is this, this pathway is affected in this bioremediation? Is a phytoremediation? That's it told, na? There's, azo reductase, that enzyme. So is there, yes, lipid peroxidation?
मैम बेसिकली अगर हम माइक्रो एल्गी के साथ ट्रीट करें, माइक्रो एल्गी ट्रीट कर रहे हैं, माइक्रो एल्गी को ट्रीट कर रहे हैं एज ए बायो रेमिडेशन, सो बायोमास डिक्रीज हो रहा है, लेकिन फर्दर साइमल्टेनियसली हमारा लिपिड्स इंक्रीज हो रहा है. लिपिड का जो पाथवे है, मतलब न्यूट्रल लिपिड्स हैं, वो इंक्रीज होंगे, जो कि अल्टीमेट पैरामीटर्स होता है बायोडीजल के लिए.
यस, ओके, राइट.
Okay, thank you, Megha. You can leave now.
Okay, ma'am. Hello, ma'am. Hello.
Yeah, Devilina, please wait.
Yes, ma'am. Have I lost my chance or something? Actually, I was supposed to join today from 12:40 p.m.
For poster presentation?
Yes, ma'am.
No, you are there. You be there.
Okay, ma'am.
Now we are supposed to start with the poster sessions, and I want to brief about the timings. Each participant will get 3 minutes. You will get the first buzzer at 2.30 minutes, and then at 3 minutes you will get the double buzzer. That means your times is up, and then the chairs will ask you the questions. Thank you.
Yeah, Momita is there? Is Momita there? Momita, Devnath? If not there, then we'll be proceeding forward. Spruti? Spruti is there? Spruti Sandeep Gaonkar? Yeah, Spruti.
Hello, ma'am. Hello. Hello.
Momita, share your poster. I hope you have heard the guidelines, right?
Yes, ma'am.
Are you facing any problem in sharing, Momita?
Yes, ma'am.
What?
Can you please send me the poster I have sent?
The poster. There is a share button, no? Just at the below. Can you use that? You have it? So we are sharing from here, Momita. Momita. You just switch on your video, at least we could see you.
Okay, ma'am.
Yes, we'll be sharing it, your poster from here.
Okay, ma'am. Thank you.
Just wait, Momita. Yeah, can you see it, Momita?
Yes, ma'am.
Yes, now you can start.
Okay, ma'am. Hello, good afternoon, everyone. So I am Momita Devnath. I am a first year BTech Biotech student. So here is my presentation. I'm going to explain We all know that fossil fuels are fading, but the real challenge is finding a successor that doesn't not use up the primary food crops needs to grow. That's why the microalgae come in, the microscopic powerhouse um, of the earth or globally, the renewable resources. We chose microalgae as they grow up rapidly or produce high lipid content and generate valuable products. So our objective is to optimize the extraction of these lipids and valuable co-products like glycerol, pigments, and create a steady carbon neutral and cost competitive biorefinery. To achieve this into the 21st century, we incorporated the latest digital tools like CRISPR modified strains to specifically boost lipid and environmental resilience. And we use AI driven photobioreactors and IoT sensors with real time monitoring. So moving to the next, the cultivation system. We used two methods in this cultivation system. First one is open pond. These are scalable and cost effective. And the second one is closed photobioreactor. This offers a controlled environment to maximize biomass. There are some factors that are affecting the microalgae while producing biofuels or byproducts like pH, temperature, light intensity, nutrient salinity, etc. So now look at the data. Our research into diverse habitats shows that marine species accounts for 53% of viable algal strains, providing a massive resource for fuel, while freshwater varieties make up 43%, which is excellent for inland cultivation. Globally distribution, if you look figure 1 and 2, we have mapped the global distribution. Europe lists with 28% documented species, followed by the Americans, which is 25%. This data is crucial because it helps us identify.
Amrita, your time is up 2.5. Only half a minute is left.
Okay, ma'am.
Finish.
Because it has
Time is up, bache. Your time is up. Just wind up, conclude.
Okay, ma'am. So combining algal growth with wastewater treatment is less cost using tools like CRISPR and AI use like IoT monitoring. The foundation of this research is built upon established literature. And finally, I would acknowledgement by University and the Department of Bioengineering of Technology University Tripura. So thank you for your time.
Thank you, Momita. So you have given a glimpse, right? That is a review which you have done, right?
Yes,
not
ma'am.
wrong. So you have given so many things, you have searched for so many things. So which kind of microalgae or algae you will be going for production of biofuel? Just name one.
Which kind of microalgae? Can you please repeat the question?
Name the microalgae or the algae which you are targeting for getting the sustainable biofuels. You have done so many literature work, right? You have studied
Yes.
so many things. Spita?
I think I use the microalgae, which is starch synthesis to divert carbon flux towards triglyceride production, triglyceride production.
My question is that what kind of biofuel you are going to produce using microalgae?
Biodiesel.
Can you name the composition of biodiesel? How much carbon chain length is present in biodiesel?
Sorry, sir. That's a really I definitely something I should. I don't work on this, so
Okay, beta, koi nahi. Okay, you can leave. And
thank you, sir.
next participant is Spurti Sandeep Kaunkar.
स्फूर्ति में ज्वाइन. Yes, Spurti, you can start. Have you switched on your video? I can't see you. Why you people are not switching on your video? Spurti, are you there? Yeah. You can start. Your voice is very low. Just remove your earphone. Just talk directly. No, we cannot hear you anything. No, nothing. You need to remove that earphone. Try it. Yes. Please move on to the next one. Miss Tina is there. Tina Sinha.
Yes, ma'am.
Okay, Tina, you may share your present poster. Yes, Purti, please share your presentation poster. Now it is there. Can oh, Tina, yes. Tina will share. Smurti is not able to share later on. Yes, Tina, start please. Don't call her. We'll take her later on. Why Smurti is there? No, we'll take next. Just say. We'll start with Tina and then we'll take Smurti. You can text there, chat. Yes, Tina, you can be there. Spurti will take you later after this. Yes, Spurti, after this. Where's Tina now? Are you speaking, Tina? Otherwise, we'll be moving to the next one. Where are you? Spurti, are you ready? Tina is there. Okay, fine. Yes, Tina, carry on.
Good afternoon, respected judges and fellow participants. I am Tina Sinha from first year BTech Biotechnology student, the Department of Bioengineering, Techno India University, Tripura. Today, I am honored to present my poster, microalgae-based carbon capture and biodiesel production. Climate change is Climate change is one of the most pressing global challenge of the 21st century, and atmospheric carbon dioxide level have exceeded 420 ppm. Therefore, sustainable and eco-friendly carbon migrants suggest and essential. In this regard, microalgae are processing due to high photosynthetic efficiency, rapid growth and lipid accumulation. Unlike terrestrial plant, they capture and convert biomass into biodiesel, offer carbon capturing and renewable energy benefits. Hence, this study aimed to evaluate microalgae for CO2 capture and assess their potential for biodiesel production. To achieve this, microalgae were grown under control lab condition with optimized CO2 light and nutrients. Biomass was harvested, lipid extracted and converted into biodiesel through transesterification. Moving to the result, biomass production increased by 35% under optimized CO2 and lipid contraction received CO2 received 30 to 40% of dry weight sustainable to biodiesel. Biodiesel yield confirmed effectively lipid conversion, domestic in the festivality.
Tina, you need to wind up. Conclude.
I conclude my presentation. Microalgae effectively capture CO2 and produce lipid for the biodiesel, and respectively a sustainable solution to climate change and future energy needs. Thank you.
sincerely thank you for MIT University to giving this opportunity and to thank you Techno India University Tripura to continue support. Thank you.
So how much biodiesel you get it from the 30% of dry cell? Yield of biodiesel from the lipid biomass. Tina,
Yes, sir.
can you tell me the yield of your biodiesel from the lipid biomass?
Sorry, sir.
Can you tell me some enzyme which can fix this carbon and which can increase the photosynthetic rate and fix the carbon from the environment? Basically in microalgae.
Many species like chlorobulguris and chlorobulguris.
Okay, Tina, you can leave and
Yeah, Tina, you can leave, please.
so next is Megha Arora.
Hello. Anji sir.
Tina, you can leave. Megha. Megha.
Megha,
Yes, ma'am. Just 1 minute, ma'am. Share kar rahi hu. Yeah. Ma'am, my screen is visible.
not yet.
Just 1 minute.
Yeah, Megha, you can start. Yes. Mute yourself.
Unmute yourself, Megha.
Yeah, Megha, then we'll move to the next. You may join later. Just check with your technical problem what you're having. Okay, just take it. Take care. You be there. We'll be listening you. She left. Spurti, call. Spurti, if you're there, please join in. Megha, unshare.
Yes.
Megha, switch on the video also. Spurti, you can join. Yes. Yeah. Yes, Spurti, you can start. Again, your voice problem is there, na? We are not able to listen you properly. I've already asked you, just remove this. Earphone hata do, we'll be able to listen you. Sometimes this is the problem which we had. Now उसको हटाओ, disconnect करो उससे. Disconnect करो वहाँ से, laptop से.
Hello, ma'am.
You have disconnected your this thing, earphone. Have you disconnected your earphone from the laptop?
Hello, ma'am. Can I proceed?
Yes, please be louder then. We are able to hear you, but your voice is very, very minimal.
Okay, ma'am.
Volume. You
Good
need
morning,
to
respected judges.
carry on.
I'm Spurti Sandeep, studying in second year BSc Biotechnology in JSS Education, in JSS Academy of Higher Education and Research. Imagine producing biodiesel that does not even just reduce emission, but also maintains the carbon neutrality throughout its life cycle. My poster presents a concept titled AI-driven optimization of algal biorefineries for carbon neutral biofuel production. We all know the urgency, rising global carbon dioxide. That level demands the intelligent and demand more than renewable energy. It demands the intelligent and also carbon aware systems. Microalgae are incredibly promising. They grow rapidly, capture carbon dioxide, and also can be converted into biofuels like biodiesel, bioethanol, and biogas, and other valuable products. But here's the real challenge. Even the algae that captures and converts the carbon dioxide, but the energy that is consumed during the cultivation, harvesting, and also the conversion can cancel out all these environmental benefits. So the real question is, are we truly designing carbon neutral systems, or are we just assuming they are sustainable? Most existing optimization models focus on maximizing biomass or lipid yield. AI application focuses on prediction accuracy. Life cycle assessment, or LCA, is usually done after the process is designing. So
हेलो
carbon neutral
निशु मेरी टर्न आने वाली है, तो है ना चली जाइए अगर मैं मतलब
Megha, please switch off your audio. Mute yourself. Mute yourself. Spurti, you don't mute yourself. I'm not talking to you. Spurti, you carry on.
I'm not able to hear, ma'am.
Then
You can
put-
continue your poster presentation. Spurti, you can continue.
She is not able to hear you. Okay, put back your earphone.
Yes, ma'am.
You can continue.
So, most existing optimization models focus on maximizing biomass or lipid yield. AI application focuses on prediction accuracy. Life cycle assessment, or LCA, is usually done after the designing of the process.
Now
And
conclude, please. Come to the conclusion.
Yeah, ma'am. So this concept embeds carbon balance directly into AI decision, making it
making it transform life cycle assessment from a retrospective evaluation tool to an active control mechanism, and introduces feedback-based carbon governance into algal biorefineries. In a world, although it is currently a conceptual proposal, its impact could be transformative. It lays the foundation for simulation-based validation, pilots,
Conclude, please. Time up.
in a world urgently seeking sustainable solution, we must move beyond simply producing renewable energy. We must design intelligent systems that guarantee carbon accountability at every stage. Thank you, ma'am.
Yes, it's over.
Yes, ma'am.
Can you name some of the microalgae used for the application in biorefinery? Can you name some of the name of algae which is used for the biorefinery application and why?
No, sir. I will study about it.
Are you able to hear now?
Yes,
Is my voice coming?
yes, ma'am.
Purti. Sir is asking about name of the microalgae which produced in the biorefineries. Name any of the microalgae.
Study about it, ma'am.
Pardon me, what did you say?
Ma'am, I'll study about it. I don't know.
You don't know. Okay, fine. So these are the review you have done, right?
Yes, ma'am. I have reviewed and I have brought up my idea about advancing the AI as a decision maker instead of just promoting the yielding,
Okay.
and a feedback loop. This is an innovative concept I have brought up, ma'am.
Which AI tool you will be using for that?
Ma'am, I have still not studied about that. But
Okay. Fine. Okay. It's good. Can leave, बच्चे.
yeah, thank you, ma'am.
Unshare, please. Unshare your slide.
Yes, ma'am. I'm reading.
Mega is there? Mega, you can share your poster.
Oh, okay, sir. Sir, my screen is visible.
Yes, it's visible. You can start your poster.
Okay, sir. Let me start. I'm Megha Arora from the Department of Life Science, JC Bose University of Science
Megha.
and the Technology. Yes,
Mega,
sir.
you can put your screen on presentation mode.
Sir, my
Slide.
screen is not visible.
It's visible, but it's not completely visible. Just put on slideshow.
Okay, sir. So,
It's fine.
visible?
Yes, yes.
Okay. My poster title is called the Fueling the Future of Metabolic Engineering of the Tag Biosynthesis in the Microalgae. Let me first to choose the microalgae, because microalgae especially choose the chlorella species because these contain high lipid productivity, rapid biomass growth, high cell content, less use of fresh water as comparison to the some other waters. And basically the you choose microalgae. Microalgae contain the fatty acid and the glycerol. Fatty acid means the standard and the saturated and the unsaturated acid, and the glycerol contain the neutral lipids. Neutral lipids, which is the ultimately parameter for the biodiesel production. These are the large number of the microalgae species contain the neutral lipid, but I choose the Chlorella sorokiniana species, which was isolate in the lab in BG11 media, and these contain 28 to 30% the high neutral lipid for, and all the algae are not suitable for the biodiesel, but the Chlorella sorokiniana is the robust strain which contain high neutral lipid used for the biodiesel production. And the in Chlorella sorokiniana, these contain the some specific genes like ACC, DGAT, and GPAT. ACC is the first committing steps in the fatty acid synthesis. If these steps are the rate determined, which control if the lipid synthesis is upregulate or not, because in the microalgae a bottleneck effect is called the, if biomass is increased, so lipid content is decreased, but if sometime lipid content is increased if biomass is decreased, so these are the bottleneck. So if the upregulate of ACC genes which contain high lipid content means the ultimate parameter for the biodiesel production. So I choose the methodology. We choose a fresh microalgae collection, which was isolated in the lab condition, and then further growth optimization, and under the stress condition like nitrogen stress, yeah, dye stress, and further to enhance the under stress conditions, the microalgae cells play
Megha,
a
you can
different
conclude
role
your
as
study.
comparison to normal conditions. So in stress
Megha,
conditions, the gene level activate, and genes are these genes are further isolate and then transform to
Megha, you can
convert into
conclude
the
your
most
study.
reproductive species.
You can conclude your study. Okay, one short question from you.
Yes, sir.
Which identification method you will use for your identification of your microalgae?
Uh, Identifications method means further use the freshwater microalgae, then further to isolate with the agar plate method.
How you can confirm that microalgae is that microalgae, particularly that
Uh, Sir, further for gene sequencing 18 sRNA.
that question, that is the question, and this answer I am expecting. Okay, thank you.
18 sRNAs.
You can leave, Megha, please.
Sneha Das, you can share your poster.
Yes, sir. Sir, can it visible?
Yes, it is visible.
So, good afternoon, everyone. Myself, Sneha Dash from the Department of BTech Biotechnology, Bioengineering, Techno India University, Tripura, and Techno India University, Tripura. So my topic is progress and challenges in lignocellulosic biorefinery for efficient biomass and deconstruction. So lignocellulosic biorefinery represents a sustainable approach for converting renewable biomass into biofuels, biomaterials, and other value-added products. Lignocellulosic biomass mainly consists of three parts. First one is cellulose. So cellulose is a glucose polymer and it is the main source of fermentable sugar. Then hemicellulose is a branch polysaccharide. Then lignin. Lignin, it gives resistance. Then it is a aromatic polymer. Then in my poster, there are 3 methods I have mentioned. First one is the pre-treatment, the core step. So pre-treatment, the core step, pre-treatment is considered is one of the most critical step in lignocellulosic biorefinery. The main objective of pre-treatment is to remove lignin structure to disrupt the crystalline structure of cellulose. And there are some methods. First one is physical methods like the milling and grinding. Then chemical methods like the alkali or acid. Then third one is the biological, that is the microbial treatment. Then second one is enzymatic hydrolysis. So after pre-treatment, enzymatic hydrolysis plays a major role in converting polysaccharide into fermentable sugar. Some enzymes are included here. First one is endoglucose and beta glucosidase. This enzyme act synergistically to break down cellulose to glucose. Enzyme inhibition, low stability, and high production cost limit industrial applications. Then the third one is
Sinaya, your time is over. Please conclude. Your time is over.
Okay, sir. So in conclusion, lignocellulosic biorefineries hold great promise for sustainable industrial development. So our future research should focus on advanced genetic engineering, robust enzyme system, and with continued innovation, lignocellulosic biorefineries significantly contribute to global renewable energy
Thank
solutions.
you, Sneha. Thank you. A quick question. Have you still working on the review part or you started research on this?
Only the review part, sir.
Okay, okay. Thank you so much. Please unshare yourself.
Okay, sir. Thank you. Sir, can I leave now?
You can now. You can leave.
Okay, okay.
Shrestha. Yeah, I have. Tripura. So next is Shrestha. Shrestha, are you there?
Yes, ma'am.
So you can start. Shrestha, you can start.
Ma'am, is it visible?
Yes.
Good
Yes.
afternoon, everyone.
Mhm.
Good afternoon, everyone. I am Shrestha Majumdar, and currently I am pursuing BTech in Biotechnology from Techno India University, Tripura. So today I will be speaking on the topic valorization of sugarcane tops through bioconversion for bioethanol and high-value products. India is one of the largest producers of sugarcane in globally. According to reports, sugarcane cultivation generates millions of tons of agricultural residues every year. So, however, from a biotechnological perspective, they are rich in lignocellulosic biomass, composed mainly of cellulose, hemicellulose, and lignin, and this make them an excellent substrate for bioconversion processes. The major challenge lies in the breakdown of this lignocellulosic structure. Then we come the methods that starts with the pre-treatment process where It includes dilute acid alkali method and steam explosion, where the rigid structure can be disrupted and enhance enzyme accessibility. Then following the pre-treatment method, we come to the hydrolysis part. It shows that converts cellulose and hemicellulose into the fermentable sugars like glucose and xylose. And these sugars can then be fermented using the process fermentation using microorganisms like Saccharomyces cerevisiae to produce bioethanol, which is a renewable and cleaner alternative to fossil fuels. Then in result part, it's showing that the compositional analysis of sugarcane tops in dry weight basis and cellulose having the range of 33 to 40% that converted into glucose, and hemicellulose having the range of 18 to 33%, and it has released pentose sugar during hydrolysis, and lignin having the range of 17 to 32%, and ash and extractivity is also having the range of 2 to
Krista, your time is up.
11%.
Please bind up. Please go with the conclusion directly.
Okay, so I conclude that sugarcane tops are the abundant and underutilized lignocellulosic biomass with high potential and sustainable bioresource utilization, and effective pretreatment and improving saccharification significantly enhance the release of fermentable sugars and improving bioethanol yield. Thank you, sir.
So, Shrestha, my question is, which pre-treatment is more effective, alkali or acid for sugarcane?
Ma'am, it's acid. Pre-treatment is more effective.
Why?
Ma'am, as it degrades the lignin parts and it converts into the simplest sugar, that is glucose and xylose.
Okay. So that means there is no need to use the microorganisms for the hydrolysis part.
No, ma'am. We can use the fermentation process to produce the bioethanol using the microorganism for the production of bioethanol.
Okay. Okay, thank you, Shrestha. You may leave now.
Okay, ma'am.
Please, Shivani. Oh. Rajat.
Hello, sir.
Are you there?
Yes, sir. Yes, sir.
Please share your poster.
Hello. Good afternoon, sir.
Please share on slideshow. Just make it slideshow. Yes, please start.
Good afternoon, sir. My name is Rajat Malik. Currently, I am pursuing my PhD under the guidance of Dr. Kushma Kumari Panda from MIT University, Noida, Uttar Pradesh. And today I present on the endophytes and their rhizosporic microbes. Abstract worldwide, approximately 19.5% of irrigated and 2.1% of rainfall land impacted by salt stress. Salinity negatively impact plant by producing iron toxicity, osmotic stress, minerals imbalances, and by disrupting various physiological and biochemical processes. Globally, sorghum is the fifth most important cereal crop, thus its salt tolerance is a matter of global concern. Furthermore, salinity changes the soil structure, inhibit root growth, reducing nutrient bioavailability, and diminishing the microbial function, eventually degrade the overall soil ecosystems. And with plant root by building an extensive mycelium network that is spread beyond the rhizosphere. In sorghum, arbuscular mycorrhiza fungi colonization significantly improve phosphorus and acquisition, which is essential for energy transfer and root development. In addition, fungal endophytes stimulate the defense mechanism by enhancing antioxidant production, which neutralize the harmful effect of reactive oxygen species. While nitrogen-fixing bacteria are mainly associated with legumes, they also play an important role in the growth of cereal crops such as sorghum. These bacteria form a loose associations with sorghum root, facilitating the conversion of atmospheric nitrogen into ammonia, supporting plant nutrition. These beneficial endophytes microbes are isolated from sorghum's germ plant and will be used to mitigate salinity stress. Impact of salinity on sorghum: decline seed germination and plant growth, reduce chlorophyll content and photosynthesis, change the soil microbial diversity. Microbial interaction in salinity tolerance. There are various microbes. First one is arbuscular mycorrhizal fungi, live in symbiotic relationship with the plant.
Please go with the conclusion directly.
Okay, then I conclude that microbial interactions substantially increase salinity tolerance. Beneficial microbes such as arbuscular mycorrhizal fungi, endophytic fungi, and plant growth promoting bacteria increase nutrient acquisition, maintain ionic imbalance, and stimulate antioxidant system. Exploiting these interactions offer an eco-friendly approach to improve sorghum productivity under saline conditions. Thank you. I have done, sir.
Time is up. Rajat, a quick question. Which sorghum species you are going to use in your research?
Sir, actually, this is a review-based paper. So,
okay. So you are not going to work on this one?
No, sir.
Okay, thank you. Thank you, Rajat. You may leave.
Okay, thank you.
As we are done with today's poster session, we'll continue with the yesterday's one. We'll start from Nikhil Dikshit and we'll continue to the Shrishti Agarwal. Thank you.
Hello. Excuse me, ma'am. Excuse me.
Yes.
Our
For today's presentation itself?
yes, yes, I am for today's also.
Yeah, my serial number is 36.
It will start from 3 p.m. Sorry, it will start from 2 p.m. We'll continue that from 2 p.m.
Okay.
Thank you.
Ma'am, my presentation was left yesterday, so can I start with the presentation?
Your roll call will be called, then you'll start with it.
Okay.
Nikhil Dikshit.
Nikhil, are you there? Then.
Hello, ma'am. Ma'am, I Rashmi. Ma'am, today is my slot. From which time it will begin?
2 p.m. will come.
Ma'am.
Okay.
Thank you.
Next is Devalina.
Yes, sir. Hello.
Yeah, please share your slide. Post
okay, sir. Okay. Sir, is my slide visible? Hello? Hello?
Yeah. Please make it slideshow. It's fine.
Sir, I have shared the slide.
Sure. We can
Is
see.
it visible now?
Yeah.
Okay.
Please.
Shall I start now, sir?
Yeah, please start.
So, good afternoon, respected judges and participants. I'm Debolina Das from Techno India University, Tripura. And today I'm going to give a review-based poster presentation on my poster titled Bioremediation of Heavy Metal-Based Contaminated Industrial Influence UIC Service here. So, industrialization has improved the human lives, but also created a serious problem, which is the heavy metal pollution and water pollution. So, the industrial influence treatments that come from electroplating, mining, and textile industries, they contain these heavy metals. These heavy metals are non-biodegradable and persist in the aquatic ecosystems and enter the food chain through biomagnification, causing serious health problems such as kidney damage, neurological disorders, and even cancer. So conventional treatment methods, these are highly costly and generate this toxic secondary sludge, which eventually increases the workload. Therefore, biological treatment methods are considered as more sustainable. So my study is based on the Saccharomyces cerevisiae, which is a non-pathogenic yeast that is widely used in biotechnology. Its cell wall is composed of beta-glucan mannobrokin chitin, and which exposes functional groups such as carboxyl, hydroxyl, phosphate, and amino groups. These functional groups helps in the heavy metal binding and hence helps in the heavy metal removal. The objective of this study is to evaluate its efficiency, that how this heavy metal removal potential of Saccharomyces cerevisiae has, and to understand its overall mechanism. And here is the reported study which describes and the methodology that how we will prepare the yeast biomass. And then second, these prepared yeast biomass are made interacted with these metal solutions. And after that, we will incubate the samples under controlled pH and temperature. And after incubation, we will estimate how much metal has been removed. And total, we will get the results. And this is the result which explains that the metals have been removed via two mechanisms, which is bioabsorption and the bioaccumulation. In the bioabsorption, the functional groups, they absorb, and the metals, they have the heavy metals bind with the functional groups, and finally in the bioaccumulation, they enter inside the yeast cell. And now this is the graph which illustrates
Develina,
and
please
explains
bind
the
the go with the conclusion directly. Go with
so
conclusion.
to conclude, the Saccharomyces cerevisiae, it is effectively can remove the heavy metals through bioabsorption and bioaccumulation mechanism, and represents a low-cost, eco-friendly alternative for wastewater treatment. Thank you for your attention, and thank you so much for listening.
Thank you, Develina. A quick question. Which heavy metals
Okay.
you're targeting for the remediation?
So we are targeting cadmium, then mercury, because mercury, they enter through the biomagnification process, so which is very important, because through biomagnification they disrupt the plants and animals by entering in the food chain.
Okay, thank you.
Okay, thank you, sir.
Okay, Develina, you may leave.
Okay, sir.
Prapti. Develina, please unshare your poster.
Okay.
Yes, sir.
Prapti, are you there?
Yes, sir.
Please share.
Sir, am I audible?
Yeah. Please share your poster.
Okay. Is it visible, sir?
Please make it slideshow.
Wait, sir. May I start, sir? Good afternoon, everyone. I am Prapti Tyagi from Department of Chemistry, SRM Institute of Science and Technology. My research focuses on developing a zero-waste biorefinery model that uses agriculture residue, such as corn cob and pineapple shells, to produce high-value biosurfactants. And the main objective of this study is to recover fermentable sugar from lignocellulosic waste, and also to utilize these sugar from rhamnolipids production. The process was divided into 4 phases, like pre-treatment and hydrolysis, sugar quantification, fermentation, and product recovery. Corn cob and pineapple shells were subjected to dilute acid hydrolysis to release hemicellulosic sugar, and released sugar were quantified using HPLC to determine various constituents like dextroxylose, arbinose, and fructose concentration. The hydrolysate was used as a carbon source for fermentation using Planomicrobium okeanocoides. most importantly from concob hydrolysate. Some as shown in table, dextroc is 35.94 gram per liter, xylose is 28.01 gram per liter, and so on. As shown in the donut chart, dextrose and xylose constitute the main fermentable fractions. Green catalyst treatment improve glucose and xylose recovery by approximately 24 to 25% compared to the conventional method. This study has various economic values like complete valorization of agriculture waste, reduced dependence on synthetic media, and also cost-effective production for biosurfactant productions. In conclusion, our work establishes a scale zero waste biorefinery platform that convert low-value biomass into high-value functional byproducts. Thank
Prapti,
you.
please wind up with the conclusion. Okay, thank you.
Prapti, my question is, the microbe that you used for your study, have you isolated it?
Ma'am, I'm just go through the review part now.
Okay, so this is the review. Okay.
Yes, ma'am.
Okay, thank you.
Thank you, ma'am.
Okay, thank you. You may leave now.
Okay, ma'am.
Next is Shakshi.
Yes, sir.
Yeah, please share your poster.
Sure. Is it visible, sir?
Yeah, please make it slideshow. Yeah,
Okay.
please
Good
continue.
afternoon, respected judges and everyone present here. I'm Sakshi Kumari from Amity University of Biotechnology. Today I'm presenting my work titled Sustainable Production of High-Value Biomolecules from Oleaginous Yeast Cultivated on Mandi Waste. India generates a large amount of organic waste, especially from vegetable and mandi areas. These mandi waste is rich in natural sugars and nutrients, but is often discarded, leading to environmental problems. At the same time, industries require sustainable and low-cost sources of valuable biomolecules such as lipids, proteins, carotenoids, enzymes, and beta-glucan. So our study focuses on converting this low-value mandi waste into high-value biomolecules using oleaginous yeast, thereby supporting waste valorization and circular bioeconomy. So let's move on to the methodology. First, mandi waste was collected and homogenized to obtain a uniform substrate. Then oleaginous yeast was inoculated into this prepared media under controlled laboratory conditions. After cultivation, the biomass was separated using centrifugation and different biomolecules were extracted and quantified. We mainly analyzed lipids, carotenoids, beta-glucan, enzymes, and proteins. So our results showed efficient growth of oleaginous yeast on mandi waste substrate. The maximum protein concentration obtained was 242 microgram per ml at 48 hours of cultivation, indicating peak metabolic activity during this period. And we have also achieved a carotenoid yield of 0.031 gram per gram and a lipid yield of 0.7 gram per gram. This finding demonstrates that mandi waste can effectively support microbial growth and biomolecule production in a cost-efficient method. The extracted biomolecules have diverse industrial applications, such as lipids can be used for biofuel production. Carotenoids are used in nutraceuticals and cosmetics. And beta-glucan and proteins have application in food, pharmaceuticals, and immune-boosting formulations. And enzymes have potential application in bioprocesses and biodegradable polymer breakdown.
Thank you. Your time is up. So a quick question. It's a review or you are still working on this mandi waste like fruit or vegetable waste?
Sir, I'm still working on this. Currently, we are at the preliminary stage.
So you are using a cocktail of waste or you are sorting out first, then you go with your methodology?
No, sir, we are using cocktail of waste.
Okay. Okay, thank you, Shakshi.
Okay, so welcome. Thank you.
Next is Arpit.
Yes, sir. Good afternoon, sir.
Please share your poster.
Sir, is it visible?
Yes, please make it slideshow. Yeah.
Yes, sir. Good afternoon, everyone. My name is Arpit Bishnoi from the Department of Biochemical Engineering, Chemical Technology from HPTU Kanpur. And my topic is, my poster is titled Advanced Materials and Technologies for Heavy Metal Remediation in Environmental Systems. So, heavy metal contamination from industrial wastewater and municipal waste is a serious environmental issue nowadays. And metals such as chromium, lead, cadmium, and arsenic are non-biodegradable and also toxic even at low concentration. And eventually, they accumulate in soil and water, posing serious risks to human health and ecosystems. And some previously traditional methods, treatment methods like chemical precipitation and filtration have limitations such as sludge generation and high cost. Also, they are low efficient at trace concentration. Therefore, this work focuses on advanced materials and technologies that can overcome these challenges. So, main remediation mechanisms which are involved are adsorption, ion exchange, photocatalysis, and electrostatic interaction. In adsorption, our mechanism is the metal ions attached to the surface of materials, and ion exchange in replacing harmful metal ions with safer ions, and photocatalysis, the light activated materials easily convert toxic metals into less harmful forms. In terms of advanced materials, this paper discussed nanomaterials and nanocomposites, MOFs, which are metal organic frameworks, which have extremely high porosity and tunable structure. Coming to my part, biochar and modified chitogens, which are sustainable and cost-effective options for wastewater treatment. So advanced technologies in this paper, which includes membrane filtration systems, electrochemical remediation, and hybrid systems which combine adsorption and photocatalysis part, and also microbial assisted treatment methods. However, there are challenges such as scalability, regeneration cost, and also performance in real wastewater system. So future research focuses on green synthesis.
Arpit, please conclude.
Yeah, so in conclusion, so advanced materials, especially nanocomposites and MOFs, metal organic frameworks, they offer significant improvements over conventional methods. So with further development and integration into hybrid systems, they have a strong potential for efficient and sustainable heavy metal remediation. Thank you.
Thank you, Arpit. Arpit, a quick question.
Yes.
After comparing your all the materials and technologies, what do you suggest? Which material and technology is good for this?
Sir, actually this is review paper, but currently I'm working from past month in for biochar production. I'm preparing a biochar adsorbent for chromium
You
removal.
reviewed the literature, right?
Yes, sir.
So after reviewing the literature, only you choose the biochar.
Yes, sir.
Okay. Okay. Thank you, Arpit.
Thank you, sir.
Okay. Okay. Thank you.
Next, Gaurang Gupta.
Yes.
Gaurang, are you there?
Yes, ma'am. Yes, ma'am.
Okay, you can start. Please share your poster.
Ma'am, is my screen visible?
No, it's visible now.
Can I start now?
In slideshow. Yes, you can start.
Good afternoon, everyone. My name is Gaurang Gupta, and I am presenting my work titled Crude Glycerol Enhanced Mixotrophic Chlorella Cultivation for Biofuel and Wastewater Valorization. So with the increasing global demand, energy demand, and environmental concerns associated with fossil fuel, sustainable biofuel alternatives are urgently needed. Microalgae are considered a promising third-generation biofuel feedstock due to their rapid growth, high lipid accumulation, and ability to grow on non-arable land using wastewater. However, large-scale commercialization is still limited because cultivation cost is high, especially the carbon and nutrient inputs cost is high. At the same time, biodiesel industries generate a large amount of crude glycerol as a low value byproduct, creating a disposal challenge. So, this study explores whether crude glycerol can be used as a low cost organic carbon source to enhance algal growth and lipid production while simultaneously treating the wastewater. So, the objective of this study was to generate an integrated economical cultivation strategy using wastewater as a nutrient medium, low cost agriculture fertilizers for nutrient optimization, crude glycerol as an organic carbon source, and
Wrong,
mixotrophic
your time is up.
cultivation.
Please go with conclusion.
Okay, sir. So, in conclusion, the wastewater supplemented with low-cost fertilizer effectively support the algal growth. Crude glycerol serve as an economical carbon source, and mixotrophic cultivation significantly improves biomass and lipid productivity. So, this integrated approach not only enhances the biofuel potential, but also enables efficient wastewater treatment, supporting a sustainable circular bioeconomical model. Thank you.
Okay, Gaurang, a quick question. Which
Yes,
species
sir.
targeting algae? Which is algae species you are going to target?
Sir, in this study, we use Chlorella species BRE4.
Why?
Sir, it is because, sir, due to its high productivity and sir, high lipid content generation, and sir, because it has high potential for treating wastewater.
Have you compared this species with other algae species in terms of productivity and the high lipid content?
Sir, I have not done that study, but I have read a previous research paper of other scientists, and due to which we have decided to use Chlorella species BRE4.
Okay, thank you.
Thank you, sir.
Okay, next, Shreya.
Am I audible?
Yeah. Please share your poster.
Is my poster visible?
Not yet. Please share. Yeah, now. Please make it slideshow.
Now this is visible.
Yeah, please. Yeah.
Okay. Good afternoon. I am Shreya Garg. My poster is on optimizing the carbon to nitrogen ratio in Chlorella species to enhance lipid productivity for biodiesel production. As microalgae are promising biofuel feedstock, but nutrient imbalance, especially carbon and nitrogen, strongly affect biomass and lipid accumulation. Chlorella species was cultivated under mixotrophic condition using glucose as a carbon source and nitrate as a nitrogen source. Two experimental setups were designed, one glucose limited and second nitrate limited condition at different ratios of carbon and nitrogen. The results show that at 29:1 CN ratio gives maximum biomass productivity and high lipid productivity. In conclusion, optimizing the carbon to nitrogen ratio significantly improves lipid productivity, making Chlorella species a strong candidate for sustainable biodiesel production. Thank you.
Thank you, Shreya.
Okay, thank you, sir.
Next, Srishti. Srishti, are you there?
Yes, sir.
Please share your poster.
Yes.
Put on slide show mode.
Sir, is it visible?
It's visible. So why
Okay.
you have chosen two-stage cultivation?
Sir, two-stage cultivation. We have used continuous two-stage cultivation because when we do two-stage cultivation, in first stage we provide a sufficient amount of nitrogen,
And in second stage, we give microalgae nitrogen limitation and provide phosphorus. So
Okay.
this makes algae accumulate lipid better.
Okay, Srishti. So you can explain about the what kind of work you have done, objective, and whatever results you got it.
So in this research, we used Chlorella HS2 species, which was which was grown in BG11 media. A bubble glass bubble photobioreactor was used, which in which unlimited conditions and unlimited oxygen limited conditions were supplied. To compensate this, phosphorus was supplied on the fifth day of cultivation. This was to optimize phosphorus concentration for its analysis in open raceway ponds and photobioreactors in three different conditions. That is with air without any nutrients, with carbon dioxide with no nutrients, and carbon dioxide along with nitrogen limitation and phosphorus and oxygen supplementation. These three treatments were followed in photobioreactors and open raceway ponds as treatment 1, 2, 3. So the best results were received in nitrogen limited, phosphorus 500% and oxygen 100% supplementation. Then these results, these optimized conditions were used in photobioreactor and open raceway pond treatments. Biomass yield that was received in open raceway pond treatment 3 was 1.85 gram per liter. That was 1.22 times higher than that of TORP2, and the biomass yield in PBR, that is the photobioreactor treatment 3, was 6.97 gram per liter, which was 1.454 times greater than TPBR2. So in overall, we can see that photobioreactor was the best conditions in which microalgae accumulated the highest biomass. Also, the highest lipid productivity was in photobioreactor 3, which was 289.6 mg per liter per day. To conclude, strategic P supplementation during unlimited second stage significantly enhances biomass and lipid productivity. N0 P500 O100 was the optimal condition for ORP or PBR scale up of microalgae. A continuous two-stage cultivation strategy avoids biomass transfer steps, making it more energy efficient and scalable. This approach is suitable for large-scale production of biofuel and reducing the use of fossil fuels.
Okay,
Thank you.
Srishti, how much is the lipid yield from your microalgae?
Sir, the lipid that we received from microalgae in the best condition was 289.6 mg per liter per day.
So it's very less, don't you think? 200 mg per liter of culture. And
Sir,
what is the
this was
productivity of lipid? Productivity?
So this is the lipid productivity that I have told you, 289.6 mg per liter per day.
Per day, okay. So how much oil you get it from this microalgae?
So that was not calculated. We have just seen the biomass and lipid productivity, and the biomass yield was 6.97 gram per liter.
Okay, okay, fine.
Thank you, sir.
From 10.
Before winding up the session, I would like to special thanks to Dr. Kirti Rani Sharma, Dr. Neha Basotra, and Dr. Krishna Gautam for their valuable time and smooth conduction of the session. It's now my privilege to invite Dr. Kirti Rani Sharma and Dr. Manoj Kumar to the stage to honor and facilitate our respected chairs for their valuable presence and guidance. Thank you, participants. The session will resume within 5 minutes. So we'll continue with the session. Distinguished delegates, esteemed scientists, honorable chair, respected faculty members, and dear participants. Good morning. I am Navarati, and it's my privilege to welcome you all to the International Conference on Sustainable Biotechnology and Bioinnovations, SBB 2026, being held from February 25th to 27th, 2026. This prestigious conference is organized and hosted by Amity University, Uttar Pradesh, in association with Biotech Research Society India, International Bioprocessing Association, IFIB, BIOP, and the Center of Energy, Environment and Sustainability. SBB 2026 brings together brilliant minds from academia, research, and industry to explore innovative solutions in sustainable biotechnology and bioinnovations. I am privileged to introduce the session chairs. Dr. Abhishek Sen Gupta. Dr. Abhishek Sen Gupta is Associate Professor at Amity University with PhD in Bioinformatics, specializing in systems biology, clinical data analytics, biology network modeling, multi-omics integration, and machine learning driven predictive modeling. His research focuses on biomarker discovery, genome scale metabolic modeling, AI-based reproductive health analytics, biomicrobiome studies, and translational bioinformatics. With extensive teaching experience in computation biology, bioinformatics, and system biology, he has supervised numerous research projects and coordinated PhD programs. He has published widely in high-impact journals, contributed book chapters with Springer, Elsevier, and CRC Press, and presented at national and international conferences. He serves as PI and co-PI on DBT and ICMR funded projects, holds multiple software copyrights in reproductive health analytics, and is a reviewer of reputed journals including PLOS 1 and BMC Medical Genomics. He is also an active member of several international bioinformatics and system biology societies. And now I would like to introduce another chair, Dr. Manoj Kumar. Dr. Manoj Kumar is a professor at Amity University with extensive international research experience, having previously served at Jawaharlal Nehru University, University of Cambridge, University of Clogon, and IBMP CNRS, France. He holds PhD in developmental biology and genetics from the University of Clogon and has received prestigious recognitions including the SERB Young Scientist Award and the Dr. D.S. Katoria Postdoctoral Fellowship. His research focuses on cancer biology, epigenetics and plant molecular biology, transcriptional regulation, and antimicrobial resistant. With the publications in leading journals such as Science, EMBO reports, Cell Reports, and Plant Physiology, he has secured multiple national research grants, serves as reviewer for major funding agencies and high-impact journals, holds lifetime membership in prominent scientific societies, and has supervised numerous PhD masters and undergraduate students. It is now my privilege to hand over the stage and to the respective chairs. I invite you to preside over the session, call over the participants, and guide the presentation and evaluation. The stage is now yours to lead the discussion, engage with our presenters, and shape the scientific disclosure of SBB. Now, I would like to clarify the time limits and the time in brief. You all will get 3 minutes for the poster session. And the first tab will be at 2.30 minutes, and then another tab will be at 3 minutes. Thank you and have a nice day.
Hello. Good afternoon, everyone, and good afternoon, participants here. We are starting with the poster presentation. The first poster presentation will be by Diksha Kumari from Dr. B.R. Ambedkar National Institute of Technology, Jalandhar, Punjab.
Yes, sir.
Are you there?
Yes, sir.
Can you? Your topic is synthesis of bacterial cellulose silica composite film for controlled delivery of biofertilizer. And I would like to tell every participant that if you could keep your background noise on a minimal level, so that we can listen. So you can display your poster.
Sir, my poster is visible.
Yes, we can see it. Just don't do that. Don't do that. You just maximize the parts. First, the upper part. You know the maximize sign and minimize sign. Zoom in and zoom out, karo. Like the on the just corner right bottom, there is a zoom in and zoom out option. Just zoom out the upper part, because if you put it in full slide mode, we can't see anything. Can you see it, Diksha? Slideshow mein? You can put it in slideshow also. Slideshow. Put it in slideshow.
Sir, 1 minute.
So we have very less time, Diksha. Can you just speed it up? So we are keeping a track of time, Diksha. It's synthesis of bacterial cellulose silica. So just can you briefly, just in 1 minute,
Okay, sir.
just tell us about your work. तो ये तो होगा ये हमें okay.
Good morning, sir.
Yes, just briefly
Okay, sir. My topic is synthesis of bacterial cellulose.
Yes. Continue.
Bacterial cellulose silica composite for controlled delivery of biofertilizer. So earlier we can use chemical fertilizers. Chemical fertilizers improve yield but cause soil degradation and environmental pollution. Then we used biofertilizer. Biofertilizer are eco-friendly, but their major limitation is poor stability and rapid loss in soil. So our objective was to develop a biodegradable and control release carrier system using bacterial cellulose and silica nanoparticle. So first we extracted bacterial cellulose from kombucha culture and purified it using NaOH and 1.5% sodium hypochlorite. Then silica nanoparticle were synthesized. After that, we incorporated silica and biofertilizer into bacterial cellulose film to form a bacterial cellulose silica composite. These composite act as a slow release carrier.
Okay, fine. I have a question. What is the advantage of this silica composite that you have synthesized on the existing ones?
Sir, silica nanoparticle improve mechanical strength and resist against pest and disease.
So why did you choose bacterial cellulose?
Sir, bacterial cellulose has a high water holding capacity and porosity.
Why?
And bacterial cellulose is a biodegradable.
I mean, all cellulose should be biodegradable. What is the feature of bacterial cellulose? How is it
bacterial
different?
cellulose is symbiotic community of
Thank you.
bacterial cellulose is symbiotic community of bacteria and yeast, and bacterial cellulose is highly pure.
Thank you.
Thank you, sir.
Next participant, Dhananjay Rathuri from Amity University. Dhananjay, are you there?
Hello, sir. Am I audible?
Yes, you're audible. We can't see your poster. Just share your poster.
Sir, is it visible now? Hello, sir. Is it visible now? My poster?
Yes. Just
Sir, should I start, sir?
आपका ही राइस पे काम
So good afternoon, everyone. My name is Dhananjay Rathodi. I am pursuing my PhD from MIT Institute of Biotechnology, and title of my poster is Agromorphological Characterization and Selection of Core Set from World Black Rice Germplasm.
Results, the graphs. Can you zoom the results? And we have your title. Just
focus on results.
Results section, yeah.
Results. Okay, sir. So in this study, I have done agromorphological analysis of 243 black rice accessions for 26 different agromorphological traits to obtain a core collection. So figure 1 is showing the cluster analysis. And in the cluster analysis, using the Euclidean matrix cluster 243 accessions into 5 different groups. And figure 2 is showing Pearson correlation. In the Pearson correlation, we have identified that yield per plant, which is a very important trait for agronomy, and it has shown correlation with 12 other important traits. And figure 3 is showing the principal component and analysis for all the 243 different traits, while figure 4 is showing PCA analysis. Yes.
Dhananjay, आप हमें just tell us your result. Don't describe your figure. Okay. That we see. What, for instance, like your figure 3, what does it show? If you have done the PCA, what does it?
Sir.
What is the outcome of that PCA?
Sir, so the PCA, sir, it is a form of clustering, but it also shows, for example, sir, the normal clustering does not show the variation within the traits, but PCA analysis.
No, sir is asking after clustering. We know it's it clusters, but after clustering, what is the result that you got?
Okay, sir.
What is your inference from that? On the basis of the PCA analysis?
Sir, on the basis of PCA, we have identified that the first 2 principal components are able to explain 48.86% of the total variation for all the agromorphological traits.
How do you say that?
Sir, sir, it's in the analysis. If I zoom the figure and if you add both the components, both x and y axis, it will be a total of 48.86% of the total variation.
Okay, please go ahead.
And then I have identified a core set of 32 accessions, and PCA was also done for those 32 accessions. for its justification, and it shows that for the core set, the PC1 and PC2 are able to explain the 55.93% of total variation, which is more than the total set. So I can say that the core set is justified, and the core set the core set covers maximum diversity of the entire germplasm.
Dhananjay, how do you differentiate your core line compared to your 250 line? What do you mean by core?
Sir, the core set is a set of accessions which covers maximum diversity with minimum set of accessions.
What are these features of diversity?
Sir, these are various traits. For example, plant height, yield per plant, panicle length, panicle weight, and flag leaf length width.
So now come back to your PCA. So which traits did correlate here? Which one do you mean?
Sir, for example, you want to see, sir, means which traits are correlated with each other?
Yes, that's what you are showing in the PCA. That's what you said, at least.
Yes, sir. Sir, we can tell that by using PCA and by Pearson correlation also. But sir, for example, that we are seeing that days to 50% heading and days to 50% maturation, they have means alignment in the.
That is not new, right? You know, it will flower. So heading date is flowering and maturation is of that. So they will correlate.
They will correlate. But sir, for example, we have seen that 1000 grade weight is on the opposite axis. Means it is showing the opposite arrow signs as days to 50% maturity. So these are negatively correlated traits.
Okay, thank you.
Thank you.
Thank you, sir.
Yes, you may leave. They're leaving, right? After their presentation. Next. Alka Mohanty.
Yes, good afternoon, sir.
Good afternoon. Can you share your poster?
Yes, sir. Yes, sir. Is it visible now, sir?
See, now it properly getting zoomed. She knows how to zoom.
Hello, am I audible to all of you, sir?
Yes, you're audible.
Okay. Good afternoon, sir. I am Alka Mohanty from Ramadevi Women's University, Odisha. Here I am presenting my work on phytochemical profiling and of Vitex spinata leaf essential oil using GCGC top MS coupled with bioactivity assessment. So as you know that plant essential oils are complex mixture of volatile phytochemicals, and they are well recognized for their diverse biological activities. Vitex spinata plant, commonly known as Laban plant, belongs to the family Lamiaceae, and these are some of the traditional uses of this Laban plant. The detailed chemical characterization and anti-biofilm evaluation of this species remain limited. So therefore, this present study aimed to comprehensively characterize the volatile constituent of this Vitex spinata leaf essential oil and to evaluate its antioxidant, antimicrobial, and anti-biofilm activity. Here is the visualization of my methodology portion. The plant essential oils are extracted through hydrodistillation process using Clevenger apparatus. Then the chemical characterization was carried out using GC TOF MS analysis. Then the antioxidant activity study was evaluated through two in vitro assays, that is DPPH and ABTS assay. Then the antimicrobial activity was evaluated by determining the MIC, that is the minimum inhibitory concentration value of the essential oil against 6 tested pathogens. And further, the sub-MIC concentration value was considered for the anti-biofilm activity. Then the result. So from the GC TOF MS analysis revealed chemically diverse phytochemical diverse profile that is dominated by sesquiterpene hydrocarbon and oxygenated sesquiterpene. A total of 177 constituents were identified, where 136 were identified for the first time in Vitex pinnata. These are the major constituents of this species, that is dermacrin D, aloe-aromatendrin, caryophyllene, alpha-cadenin, and gamma-murulin. In this GC TOF MS technique, the separation of the constituents were carried out based on their volatility and their polarity in the first and second dimensional column respectively. Due to the enhanced resolving potential of the two used columns, several co-eluting components obtained from first dimensional column were clearly separated after elution through the second column. You can see this reconstructed chromatogram. These 4 compounds, alpha-murulin, beta-salinin, cuperin, and hexadecan, these 4 compounds are co-eluted on the first dimensional column at same retention time, that is 1301.5 second. But due to their variation in polarity, they are separated in the second dimensional column. So this two-dimensional chromatographic approach significantly improves the compound resolution, and it also enables us to detect the few minor components that are present in essential oil that are often overlooked in conventional GCMS analysis. Then the antioxidant activity study. The essential oil demonstrate a notable antioxidant activity with IC50 value of 1.1 mg per ml for ABTS assay and 5 mg per ml for DPPH assay, indicating the effective free radical scavenging capacity of the essential oil. Then the significant antimicrobial activity, antimicrobial effect was also observed against all the tested strains with the lower MIC value of 12.5 microliter per ml for Acinetobacter baumannii, E. coli, Enterococcus faecalis, and S. aureus. This variation in the inhibitory effect of the essential oil on these bacterial strains might be attributed to their composition of their cell wall. Furthermore, the essential oils at its respective sub-MIC concentration, that is half of their MIC concentration.
Yeah.
Half of their MIC concentration.
Time up.
Okay, sir, just 2 minutes, sir, please, sir.
Already, yeah, sorry. Your time was 3 minutes, right? Okay, so I have just a quick question.
Yes, sir, yes, sir.
In anti-biofilm activity.
Yes, sir.
Is it biofilm activity or anti-biofilm activity?
So
and what is biofilm you mean?
So biofilm is a structure that are formed by microorganisms to protect themselves from any stress, like any antibiotics or any adverse condition. So this is the anti-biofilm activity. Means, sir, I want to check whether my essential oil sample is able to inhibit that biofilm formation of that.
How did you do that?
Sir, I did this by crystal violet staining method. And sir, in this study, I got Klebsiella pneumoniae showing highest inhibitory effect of 84%, about 84% of inhibition as compared to other strains.
How
And
did
sir,
you
I also
calculate inhibition?
so there is a formula where we take control. We take a control sample, then we deduct it from our this microbial sample. Then so there is a formula, sir.
Thank you.
Okay, sir. Sir, here two are my publications, sir. Out of this
Thank
work.
you. Thank you.
Okay, thank you, sir, for your attention.
एक ही काम है, थोड़ा सा बेहतर हो सकता है, पर
Next. Riddhima Singh.
Good afternoon, everyone.
Can you
Yes, sir.
poster?
Yes, sir. Just a second. Sir, is it visible?
Yes.
Okay. Good afternoon, everyone. My name is Riddhima Singh, and today I'll be presenting my poster on the topic ITS sequence-based phylogenetic relationship of
Please zoom.
Sorry, sir?
Can you
Please.
zoom it out, please?
Just
Yeah. Go ahead more a bit. Two results. Yeah, that's okay.
okay. Okay. Sir, should I begin directly with results or should I give some
Yes.
background information?
Yes.
Okay. So in this study, what we have done is we have used nuclear ribosomal internal transcribed spacer sequences to resolve the phylogenetic relationships among the cultivated chickpea, which is Cicer erythanum, and its representative wild Cicer species. For that, we used the ITS1 and ITS2 regions. The methodology included the total genomic DNA isolation of the 10 species of Cicer using modified CTAB method. Further, after DNA isolation, these were subjected to PCR amplification. The ITS regions, namely ITS1 and ITS2, were subjected to PCR amplification using specific primers, namely ITS1 and ITS2. Sorry, for ITS1, ITS2 and 5 were used, and for ITS2 sequence, ITS3 and ITS4 primers were used. After PCR amplification, we used agarose gel electrophoresis to size separate the amplified products. After that, the cloning and sequencing of these ITS regions was done, and further analysis of data was done using sequence alignment was done using Clustal X software, and then phylogenetic tree analysis was done using PAUP software. That means phylogenetic analysis using parsimony. So what we got was that the ITS1 and ITS2 lengths in Cicer species ranged from 235 to 239 base pair in ITS1 and 212 to 213 base pair in ITS2. The variation in GC content among ITS regions ranged from 50 to 52.3% in ITS1 and 48.8 to 50.4% in ITS2. In the phylogenetic tree, two major clades were observed. The clade 1 comprised of Cicer erythanum, Cicer reticulatum, and Cicer echinospomum. The clade 2 comprised of Cicer judaicum, Cicer bijugum, Cicer corasanicum, Cicer cuneatum, and Cicer microphyllum. It was also seen that Cicer yamashita and Cicer pinnatifidum were present as separate branches in the tree, and all the sequences of the Cicer species.
Riddhima,
Yes,
are you a PhD student?
yes, sir.
Okay.
Okay.
So is it a part of your of any of your of your thesis work, right?
Sir, it is not part of my thesis work, but I have been involved in this work as well.
Okay.
Okay. So all the sequences of Sisal species.
So how do we do you know the method or the algorithm behind parsimony, how parsimony tree is formed?
Yes, sir. We used software named PAUP for the phylogenetic tree construction using the parsimony approach.
No, I'm asking what is the algorithm? How these clades were formed, or how this scoring was done? So what is the methodology or the algorithm behind it?
Sir,
What do you understand by consensus parsimony tree?
Sir, the scoring was done using the binary method of 1-0.
And what happens after that?
So after that, the clades were formed, the each clade represents the closest species to the cultivated species that we were comparing to. We used Vishya cracca as an outgroup for the study. So it was found that Cicer reticulatum and Cicer echinospermum were very close to the cultivated species Cicer eritinum. That means they fall under the primary gene pool and are readily crossable. So the wild alleles, the informative or useful wild alleles of these species can be used for breeding purposes to introgress in the cultivated species Cicer eritinum.
So how do you think it can be used for breeding?
Sir, the phylogenetic relationship among the different species of the genera Cicer give us the information of the closest progenitor to the cultivated species.
But in the breeding, it needs to go through with the meiotic crossover, right?
Yes, sir.
So do you think it can easily cross over? What is the distance from the centromere, etc.? Do you know anything?
Sir, what I understand is, yes, it can be easily crossable because it falls under the primary gene pool. Usually the species that fall under the primary gene pool share common progenitor and can produce fertile offsprings.
No, but then if they are too close, they cannot go for crossing.
But there are variations. That is why there are two different species.
Okay, thank you.
Okay, so thank you.
Next, Sagar Ruhal.
Good afternoon, sir. Am I audible? Good afternoon, sir. Am I audible? नहीं हो रहा है कनेक्ट. Hello, good afternoon. Good afternoon, sir. Am I audible?
Yes, you are audible.
Yes, sir. Myself, Sagar Ruhal. Title of I am PhD scholar at MIT Institute of Biotechnology.
आपके दो सिस्टम्स ऑन है क्या?
Yes, sir.
एक ऑफ कर दीजिए।
नहीं हो रहा।
He must have not joined, but he has opened 2 devices. Yeah. So he shouldn't do that, no? That's why the echo is coming.
Hello. Hello.
Yeah.
Good afternoon.
Wait, just mute one of your devices voice speaker, the mic. एक का माइक ऑफ कर दीजिए आप, और एक
Yes.
से बात हाँ अब
Yes.
हाँ
Yes. Title of my poster is Leaf Surface Microstructure as an Adaptation in Stress Resilience of Lentils. Lentils, commonly known as Lens culinaris, that is basically the masoor dal. It is Rabi season pulse crop basically, which is being germinated across the globe. It is basically rich in proteins, that is 25%, vitamins, minerals, carbohydrate, and certain micronutrients such as magnesium and iron. Lentil, because of high nutrition and medicinal value, it is being consumed in our food dietary as a food dietary component. And this makes fit for cultivation in areas as it have drought tolerance ability, so it can be cultivated even in limited water availability areas. Lentils also play an important role in as a rotation crop in enhancing fertility. And due to adverse climatic condition, the need of plant-based food is increasing, which necessitates the need for developing climate resilience in lentils. And various surface microstructures such as trichomes and stomata play important role in combating tolerance to various abiotic and biotic stresses. And lentil also utilize such categories like variation in trichome and stomatal characteristics. And the potential benefit of utilizing microstructural traits might unravel the stress resilient nature of lentil. The present study explores 5 microstructural traits, that is trichome length, trichome density, stomatal density, stomatal index, and epidermal cell density in 5 cultivated accession of lentils belonging to 3 different agroecological zones, 2 from northern temperate, 1 from Mediterranean, and 2 from South Asia, which reflects the adaptability of cultivated lentil in various different climates. For this, we have used the methodology for first we have germinated the accessions, and after germination, we have collected the leaf samples for analysis of various leaflet microstructure. We have used 2 strategies like preparation of slide using epidermal peel, followed by mounting on the slide with droplet of water, and studying the various stomatal and trichome traits. Like stomatal trichome length was measured using the ImageJ software, while other microstructural traits were calculated through in particular slide sample
So
itself.
time is over.
Okay, sir.
Yeah. So just quick questions.
Yes.
What is trichome?
Trichomes are the hair-like appendages which are basically present on the surface of leaf.
Is it a cell or a tissue?
These are the epidermal leaflet structures, sir, and these play important role in providing tolerance and play important role in protection to the plant, provide tolerance to various abiotic and biotic stress.
So this is what I am asking. Is it a cell or a tissue? Is it single cell, multicellular structure? What kind of structure is it?
Sir, trichomes are of 2 types basically. Yes, sir.
Yeah, carry on.
Yes, sir. The non-glandular trichomes basically are simple unicellular hair-like structure. They are having tapered ends, and same is shown in figure number 1D.
So how do you explain that they can help in any kind of abiotic stress?
Yes, sir. Sir, like higher, just our study identifies the basically the variation basically more number of diversity in particular trichomes stomatal characteristics. So higher values, yes sir. So higher values of higher values of trichome length, epidermal cell density, stomatal density indicates that these these provide greater tolerance in combating tolerance to various abiotic stresses, sir.
Okay, thank you.
Yes, sir. And also for the first time, we have identified the glandular trichomes through this study. However, glandular trichomes play an important role in storage and absorption for the different structures. Thank you, sir.
Thank you. First year student. First year. Yeah. Maybe won't be lab then. There another part time. नहीं full time है तो full time my god सर क्या करवाएंगे ऐसा सर आते नहीं है ना अगर मैं नहीं मैं भी वही सोचा बट अब वो नहीं कराते अभी वो शिक्षिका मैम वगैरह कराती है yeah.
So next candidate is Rusha Kar.
Good afternoon, sir.
Yes, Rusha. You can share your slide. Sorry, your poster.
Yes, sir.
Yes, and tell all the candidates that there will be negative marking from now. वो तो मैंने वो तो उसके लिए है बट इसके लिए भी नेगेटिव मार्किंग होगी हाँ हाँ ये तो उसके मतलब उसके लिए किया गया था बट इसमें भी अब नेगेटिव मार्किंग कर देंगे
Candidates, I just want to tell you that after 3 minutes, the marking will be negative. So don't exceed your time limits and please follow the time limits rules. Just focus on your results only. Just tell us the result.
Hello, sir.
Yes.
Hello.
Can you zoom the result section little bit? Zoom out. Results.
Yes, sir.
Yes. Now tell what have you done and what are the results and what is the inference that you have got.
Okay, sir. So actually, my work is yet to be completed. So I concluded the result based on the phenotypic characters of crops and the from the review of literature. So, sir, I took the seven species of lentil. Among these, Lens culinaris is the cultivated species, and rest are the wild species. And we impose drought stress at 40% and 25% field capacity, heat stress at ambient plus 4 degrees centigrade during flowering up to pod maturity, salinity stress using NaCl levels at 50, 100 and 200 millimolar concentration, and aluminum toxicity through AlCl3 treatment at 150 millimolar concentration. So we got the result that under drought stress, wild species exhibit delayed flowering, reduced transpiration rate, and notably deeper root system compared to cultivated lentil. Under heat stress, biomass flowering and pod set were reduced across genotype, but some wild species maintain comparatively better reproductive stability. Salinity and aluminum toxicity reduce nutrient uptake, root and shoot dry weight, and ultimately seed yield. These stresses also influence various physiological and biochemical processes such as organic acid excretion, ROS detoxification, and hormonal signaling. But the wild species appear to be active this protective mechanism more efficiently. So we concluded that the wild lentil species possesses diverse adaptive traits that make them valuable genetic resources for improving abiotic stress tolerance. And these crop wild relatives can serve as trade donors in breeding programs aimed at developing climate resilient lentil varieties.
देखो ये चीज तुम्हें बताओ ये रिजल्ट देख के हम कैसे रिजल्ट्स आर एग्जैक्टली कुछ तो कुछ नहीं पड़े बिकॉज दीज आर वेट लैब एक्सपेरिमेंट्स राइट दिस कैन नॉट बी डन दीज आर नॉट बायोइन्फॉर्मेटिक्स फिर भी हो जाता है और हमने 2:30 बजे के बाद शुरू किया है मैडम हाँ नहीं मतलब हाँ ठीक है ना ओके ओके we'll keep checking yes
yeah I have a simple question.
Yes, sir.
How will you use wild lentil for stress tolerant, wild variety for stress tolerance? The yield will be too less in wild varieties anyway.
Yes, sir. But there is a paper in drought stress. They identified that that the wild species are more tolerant than the cultivated one. So that's why, sir.
I ask you, yeah, that is published. So how can you do that? How can you make use of it?
Sir, our work is in heat stress, sir, actually.
Okay. So what will you do?
Sir, we are giving heat stress at flowering stage, like we'll giving heat stress ambient plus 4 degrees centigrade, and we'll keep that pot up to that pot maturity. After that, we'll do the transcriptomics. But sir, as we didn't get the sample yet, so I can't present it here.
Thank you.
Thank you, sir.
Next, Carlo.
Next is Pritam Das. Next is Pritam Das.
Yes. So is my screen visible?
Yes. So just tell what have you done, your results, main results, major results, and the key conclusion from the results.
Okay, sir. So my name is Pritam Das. I am from Techno India University, Tripura, currently pursuing my BTech in Biotechnology. I am in first year right now. So my project is on plant-based alternative proteins from the next generation functional food, a step towards circular bioeconomy and sustainability. So we all know that animal proteins are little bit of costly and more that much environmental friends according to plant. So many plant sources like fruits that contain 8 to 10% of protein, like legumes that contain 45 to 50% of protein, and vegetables and many other products like soya, they contain a high amount of protein, and also fiber, which is lacked in animal proteins. Fiber is also important for our gut health and our digestion. So plant-based proteins will be a greater and best alternative for the next generation protein source. And we can also show that the graph show that the plant-based protein market has increased a very significantly from 2020 to 2026, and also the future estimate is almost 7.8% for the growth from 2026 to 2023. And the graph is based on review literature. So, and it also help us. How can plant proteins help us in the circular bioeconomy? So plants proteins that are made mainly use raw materials which are byproducts of agro-based industries, like raw products of like many residues of cereals and legume plants, which can be used for raw material to make protein using many other methods of extractions like conventional methods and novel methods, and then drying and processing to protein. So it helps us in preserving our environment and also make a sustainable protein source for the next generation. Thank you, sir.
Thank you. I have one question. You said lentils contain 50% protein, right?
Yes, sir.
Which lentil?
25 to 50% protein, sir.
Which lentil?
Sir, like rajma and dals. But one of the problem is that their protein amino acid profile was not complete. So we have to mix many other lentils also with them. Also nut, we have to add nuts with them.
Just to correct you, lentils usually never go beyond 25% protein. Thank you.
Sorry, sir. I will be sure. Thank you, sir.
Yes, Sonali Kashyap. So next is Srishti Tiwari, Amity University, Noida.
Good afternoon, sir.
Good afternoon. Please share your poster.
Yes, sir. Just a second. I hope it's visible. Hello?
Please zoom in.
Hello?
So zoom in because your format is looks to be little different. Yeah. Yes, yes, yes.
So good morning all. Myself Srishti Tiwari from Amity University, Noida. My research is my poster presentation is on the topic machine learning for early cancer biomarker discovery. So breast cancer is one of the major causes of...
Can you just scroll your are you the single author or you have another author co-authors also?
Yes, sir.
Can you show it to the top of your poster? No, like no, just scroll to. You are the single author, right? No.
Yes, sir.
No, there is Amit Chaurasia and all. Okay.
Yeah.
But why did you write only Srishti Tiwari? I don't know. Yes, continue.
Yes, sir. So breast cancer is one of the major causes of death among women. We know that early detection can greatly improve survival, but traditional diagnostic methods sometimes fail to detect cancer at the molecular level. So in this study, I used gene expression data and machine learning to find important genes that can help in early detection. And for this, for this research, I used RNA sequence gene expression data from the TCGA BRCA dataset. This dataset included gene expression or information from the tumor samples and the normal samples. And then first I cleaned the data sets by removing the errors and handling missing values. And then I selected important genes using statistical methods like t-test and ANOVA, then machine learning models like Lasso and random forest. Then this helped me reduce thousands of genes to 100 important genes. Then I used PCA and TSNE to visualize whether the tumors and normal samples could be separated. Then finally, I trained the machine learning models like support vector machines and random forest and neural network to check the efficiency of the results. Then the visualization that I showed are showed that the clear separation between the tumor and the normal samples. Among all the models, the random forest performed the best, giving about 95% accuracy. And this means the model was very good at distinguishing cancer and normal samples. Also, the selected genes were linked to important cancer-related processes. So, by concluding this, the machine learning combined with This presentation shows that the machine learning combined with
Your time is over. Please conclude.
Sorry.
Time is over. Time is up.
Okay. So the machine learning combined with gene expression data that can help identify cancer biomarkers. Thank you.
So what was the gene expression? From where have you taken the gene expression data?
So TCGA.
And just tell me, frankly, how much time did you take to perform this whole experiment? Did you get my question? How much time did you take to perform this whole experiment? I think she's lost.
Audible? Are we audible?
Yes, sir. Yes, sir. Now you're audible to me.
So how much time did you take to perform this whole experiment? Taking the
So
data?
it took me about 3-4 days to do this experiment.
You are a undergraduate student?
Yes, sir.
So these biomarkers that you have identified, how could you confirm that these biomarkers are right or wrong? But that means how do you validate it? This is a black box. It's a black box output, right? So how do you validate? How do you confirm that this is correct?
So the genes that were the gene expression data that was collected from the database. When I trained the model, it made a comparison between the genes, the tumor cells and the normal cells. So that's how I got to know that these are the correct data sets. And
thank you. Thank you.
Thank you, sir.
Srishti.
Yes, sir.
So next is Priyanka from BITS Mesra, Ranchi.
Oh yeah, a very good afternoon to everyone. I'm audible, sir.
Yes, you're audible.
Just a minute, I'll share my poster.
Share your poster.
Is it visible now?
Yes, it's visible.
Okay, thank you. A very good afternoon to everyone. I'm Priyanka from BIT Mishra, pursuing my PhD under Dr. Ramesh Chandra. And my focus of study is here the sequence to kinetics pipeline using the machine learning strategies for high throughput prediction of KCAT and KM values. The KCAT is the turnover number and KM is the Michael-Menten constant, which are very much useful in analyzing the enzyme catalytic efficiencies. So previously, traditional work modules had involved many of the laboratory tasks where it was time consuming and very costly. So the generation has moved now towards the machine learning as well.
Move to the result section.
Learning from the prediction.
Move to the result section.
Yes, sir. My methodology exactly compiled with learning and providing the training with the data sets of sequences and the sequences of enzyme and substrate, and then analyzing it through the algorithms. So here are my results, which leads to understand what exactly the output was. Firstly, analyzing the data set which I used, it was prepared from using Brenda and UniCAP. So the data set which I used has a frequency frequency class distribution of different EC classes of all EC6 classes incorporated. The distribution is being shown over here, and as well as the enzyme sequence length distribution across the data set has been presented here in the B plot, where highest frequency of enzymes are available in between the length of 300 to 400 sequence length. Next, moving to the next subsection of my result, where the representation and learning how it went through. Exactly, the enzyme and substrate get bands to bind itself to produce to a product, and via representation module, it's very important that how you vectorize it to the computer learning. Like before giving the training, the vectorization form is important. And
you can see the result. I have a question.
Yes, sir.
Proteins, there are different proteins from different protein families.
Yes, sir.
Right, and their sequences vary.
Yes,
how do you deal that problem in this particular pipeline?
Sir, actually, in my pipeline, we have focused on this section only what you have mentioned. It's like we have identified that the sequence length varies. So we have implemented a new novel module, that is segmentation. So we segmented the sequence. It's on basis of 200 sequence length. So we segmented each sequence, and then in turn we have also divided the 4 more positions. That is the 4 of the amino acid codes have been taken, and then we have analyzed each sequence on the basis of that. So the attention has been provided, like the attention is being implemented over here, and it will give precise attention score to each of the mer position of the sequence. So even
do you think there will be bias in if you follow this process?
No, sir, because I think like we have taken all the EC classes, and as well as we have dealt with the different sequence length variation. Even it's our module is representing the performance is very well, even for the sequence length of 50 amino acids, and the last two up to 1700 to 2000 sequence length. So the score metrics were very good, like R square value and the RMSE, that is the error loss, was very less, minimized as compared to some of the very known state of the art approaches till now.
A very simple question for you.
Yes, sir.
If you have done the enzyme kinetics
with
the enzyme and substrate, could you model with any known enzyme substrate here?
Yes, sir. Like in my biological insight section, I have represented my some of the set of the examples. Like I have worked using, actually my PhD work is to identify the bottleneck enzyme of the TIS pathway. Of these, the values are still not known for many of the enzymes which are participating in TIS, that is terpenide indole alkaloid pathway. So for that, I have opted the computational module. And from there, I got to know of the KCAD values of some of the enzymes which are very well they are coming as bottleneck for me, and even as well as I have co-reviewed it right now with my literature. So it's same, the stictocidin and stictocidin glucosidase, which are representing as a lower COCAT performing lower catalytic efficiency performing enzymes in the pathways.
So what's the feature did you put here to identify the substrate?
Sir, it was the smiles. For substrate, it was the smiles, and for enzyme, it was the sequence. Till now, in this model, we didn't incorporate 3D structure, but hopefully in future, even we will incorporate 3D and as well as some physiochemical properties like pH and temperature ones.
Thank you.
Okay. Thank you, sir. Can I leave from the session, sir?
You can leave, Priyanka.
Yeah. Thank you, sir.
Lavanya?
Yes, sir.
Can you
Good afternoon, everyone.
Share your poster, please?
Yes, sir. Is my screen visible?
Can you please zoom?
Yes, sir.
Directly come to the results, please.
Yes, sir. So our study was on the topic bioethanol production under high sugar stress levels. So what we did is we tried to carry out the experiment by varying the dextrose concentration in our fermentation media, which was from 2% to 40%. And then we collected the samples at different time intervals, like 24 hours, 48 hours, and then 72 hours. And then we performed the chromic acid assay for the quantification of the ethanol produced. So here is the photo of the 96 well plate in which we carried out the chromic acid assay. So basically, the blue color indicates the oxidation of the ethanol by chromium ions. So what we saw was that maximum ethanol was produced between the 11% to 25% concentration, as you can see in the graph. The blue lines represent the ethanol at 24 hours, orange ones at 48 hours, and the blue and the green ones at 72 hours. So basically, among the different timestamps, we observed that most of the ethanol concentration was found out to be at 48 hours. So this was the result of our study.
Okay. What do you think is the limiting factor here to produce efficient
So basically, there are different parameters. Like first is the high sugar levels, which becomes toxic to the Saccharomyces cerevisiae itself after a particular concentration. And another factor is the ethanol concentration in the media which is produced. Because as we know that ethanol is somewhere toxic to any organism beyond a certain level. So these are the different parameters, but as of now, we try to target only the high sugar content.
Okay, thank you.
Okay, sir.
Next.
Next candidate is Yuvika Jain from Amity University. Please show your slide.
Sir, good afternoon. Am I audible?
Yes, you're audible.
So good afternoon, everybody. I'm Yuvika Jain from Amity Institute of Biotechnology, and today I'll be presenting our work on enzymatic strategies to mitigate BNE of flavors and plant-based proteins. We are currently facing a plant protein paradox. While consumers are increasingly adopting plant-based diets for health and sustainability, acceptance remains limited due to persistent off flavors, described as BNE, grassy, or bitter. Despite strong intent, negative sensory experiences often prevent continued consumption of plant-based proteins. This off flavor originates from a biochemical pathway. This is the biochemical pathway. Plants naturally contains polyunsaturated fatty acids such as linolenic and linoleic acids. These fatty acids are highly susceptible to oxidation. Logs 9 and 13 catalyzes the insertion of oxygen into these fatty acids, producing hydroperoxides. These intermediates are then cleaved by hydroperoxide lyase to form volatile aldehydes, particularly hexanol, which imparts this characteristic grassy and raw bean-like aroma. Existing mitigation strategies, including anaerobic processing and thermal treatment and microbial fermentation, face limitation related to scalability, protein quality, or protein process variability. This highlights the need for a more targeted and mechanistically informed solution. Our work focuses directly on inhibiting the logs-driven pathway through enzyme-based intervention rather than broad processing modifications. Structurally, we identified conserved catalytic features of logs, including the non-heme iron center, coordinated by histidine residues, and conserved motifs that define the active pocket. These structural insights form the basis of our strategy design. This is the target methodology. Using in silico structural analysis and docking approaches, we mapped potential protease interaction sites within the LOX catalytic region and proposed a target protease-mediated inactivation strategy. Based on these insights, we developed a preliminary enzymatic framework combining selective LOX hydrolysis and aldehyde detoxifying enzyme systems to modulate flavor formation at the pathway level. Our initial findings suggest that targeted trypsin-mediated hydrolysis can reduce volatile aldehyde formation by approximately 35% under mild conditions, indicating that enzymatic modulation can mitigate off flavors while preserving protein functionality. Importantly, this works remain in its early stages, and ongoing efforts are focused on experimental validation and optimization. Overall, our study proposes a structured, guided enzymatic approach that shifts flavor mitigation.
Yuvika, your time is up. Please
Time
conclude
is
with
up.
presentation.
Thank you,
So,
sir. That was it.
so the you have done the in silico part or the wet lab part also?
So we have started with the wet lab and completed the in silico part and got that trypsin is the most effective protease for this enzyme.
Okay. So, like, no, how do you want to take it for further?
Further, we will be using protease solution and of trypsin, and then we will test it on the pea protein and soy protein, whichever will be easily available. And after reaction, we will use a DNPH assay, which is 2,4-dinitrophenyl hydrazine, which detects the aldehyde and ketone. If we will be successful, then the study will be experimentally validated.
So, are you aware that there are some AI based? I'm just telling you that we have done such work that AI based formulation, predictive formulation also gives a lots of success in such processes for optimizing the protein texture and since you're working on food flavor profiles, so this is a suggestion for you that you can try that also
Okay, sir. Thank you.
for optimizing it.
Okay, sir.
Thank you.
Shall I stop sharing?
Yes. Next is Aksa Khan. She's not there. Ayashi Bharadwaj. Okay, Rahul Sharma from GNDU, Punjab.
Hello.
Yes, Rahul. Can you share your poster?
Wait, sir. Hello. Good afternoon, sir.
Afternoon.
Hello.
Yes, good afternoon.
Am I audible, sir?
Yes, you're audible. Just tell your topic and brief introduction and go to the result section.
Okay, sir. Actually,
Why
I'm Rahul
slide
Sharma.
is okay? I think it's folded or probably. अभी लग रहा है. Okay, go ahead.
Okay. Good afternoon, everyone. I'm Rahul Sharma from Guru Nanak Dev University. Today, my poster presentation topic is designing of the bacteriocin-derived synthetic peptide as anti-cancer agent. So, as we all know, that the cancer is a leading cause of illness and death worldwide, and many current chemotherapeutic drugs cause serious side effects due to their non-selective actions on normal cells. Thus, there is a urgent need to look for the alternative way other than the chemotherapeutic drug. So, bacteriocins are positively charged
empathic peptides that are emerging as potential anti-cancer agent because they can target cancer cell more selectively than normal cell. So in this study, we use the computational tools. Computational tools were employed to design the short active peptide motifs from the bacteriocin gene of the probiotic Enterococcus ESMR 18. So two synthetic peptides were up 10 amino acid length, and second is about 12 amino acid were synthesized and evaluated for the cytotoxicity against the multiple cancer cell lines. Both these two peptides were shows a good cytotoxicity selectively against the breast cancer cell line, and further the fluorescent microscopy also done to evaluate the way of mechanism of the anti-cancer nature, like how they do the anti-cancer activity, like through nuclear condensation or nuclear fragmentation, or whether through separated gates. So overall, this study include that these synthetic appear to be
Your
anti
voice is breaking.
cancer therapy, and
You are not audible.
should need to be further evaluation for the in vivo evaluation. So mostly the methodology that we was performed, followed by identification and selection of the bacterial gene. Am I audible, sir? Rejoin, sir.
Yes,
Hello.
yes, you're audible, but your unfortunately your time is up.
Sir, just give me just 1 minute. I will conclude all these things. Just 1 minute.
Yes, continue.
Okay, sir. So mostly what we did, we start with the like whole genome sequence of the enterocin strain, and after that we predict the bacteriocin genes in the whole genome sequence using the various online softwares, and after that we predict the bacteriocin gene structure through the Swiss homology modeling, and after that we predict the bacteriocin sequence from the blast X, and we take that bacteriocin sequence and put it into the anti-CP, that is also an anti-cancer predicting web server, and
So
that web
one
server
thing,
designed a
listen, Rahul. Have you
Yes, sir.
done the the toxicity or the off-target interaction analysis in this?
Actually, sir, what I did, I using the web server and these two web server, and one is ToxinPred that predict about the toxicity, and after that about the anti-cancer activity, I use the different three
But that
these four
result you
web
have not
servers,
shown.
and so actually, sir, that is the these are the prediction, and after that we design that these synthetic peptide like wet lab results about this of these synthetic peptides. So both the peptides show the good anti-cancer activity against the breast cancer cell line, like against the
Thank
MCF,
you.
as well as the
We got your
triple.
work. We got your work. What you have done. Thank you so much, Rahul.
Okay, thank you, sir.
Next is Prabha. Prabha.
Thank you,
Yes, sir. Can you hear me?
Volume.
Hello.
Yes. Prabha, share your poster.
Yes, sir. I am sharing. Sir, somebody else is sharing. That's why my screen is not able to share. Now, can you see the poster?
Not yet. But that
Can
person has to join. Otherwise, how can we see the poster?
Am I?
Of course,
Is
the
my
other person, if sharing, that person has to join the meeting. Did you get my point?
Yes, sir. Is my screen now visible?
No.
Till now.
Yeah, you have to share the screen.
Yeah, I have shared my screen.
No, it's not visible. You have not shared.
Is it now visible?
No, we can't see anything.
But my screen is showing my poster.
Yes, now it is
Okay, I would
visible.
request.
Just zoom it, zoom it, zoom it, zoom it.
Okay. Is it now visible?
Yes. So just tell the title of your work, a little very crisp introduction
Okay.
and results. Just results. Explain the results and the outcome.
Okay, sir. Good afternoon, sir. My name is Prabha Bhattacharjee and I am from Amity, Lucknow. And my poster is titled Structural and Functional Characterization of Alpha Synuclein and its Phylogenetic Analysis. Actually, we have taken alpha synuclein, which is a very small neuronal protein of about 140 amino acids, and it is mainly responsible for it is present at the presynaptic terminals, and its aggregation at that point is responsible for Parkinson's disease, which is a neurodegenerative disease. And this protein consists mainly of three regions: N-terminal region, and NAC region, means non-amyloid beta component region, and a third is the C-terminal region. So in this thing, we have just taken the human alpha-synuclein as our base, and from that we have considered 30 amino acid, means alpha-nuclein sequences from 30 different species, and then we have made a phylogenetic tree according to that. The main objective of this study was to analyze the evolutionary conservation of this protein across different species and to construct a phylogenetic tree from that, so that we can understand what is the evolutionary relationship and how the sequences of the protein has been conserved and what are the variable sequences. Okay, for this we have taken the sequence from NCBI in FASTA format, and we have used Mega 12 and Cluster W in that Mega 12 software, and we have used this to prepare a phylogenetic tree using the UP geometry and using the Poisson's method. And we have used the Poisson's method to rectify the evolutionary significance and all that. And this means this tree proves that proves the well-known fact, the evolutionary relationship between the mammals and those who are not from that category, from the other taxa. Their tree or their branches are far away from the mammals. This is the thing that we have studied, and from the alpha-synuclein protein we have used this. Thank you, sir. Is there any question? I tried to make it as short as I can.
You are a undergraduate student?
No, sir. I am now pursuing MSc. Means I've completed my undergraduate. I am in the second sem of the MSc. Just entered the second semester.
Okay. So do you have any idea about aggregation propensity in phylogenetic tree analysis?
No, sir. Only I have understood that if it aggregates, if the protein aggregates at the presynaptic terminals, then it is a major cause of the disease, the Parkinson's, well-known Parkinson's disease.
So how those conserved region domains can be correlated with these aggregates?
Sir, the conserved region, actually the conserved region is the N-terminal portion, okay? And this shows the evolutionary relationship. And the portion responsible for aggregation is the NAC region, which is the full form is the non-amyloid beta component region, which occurs at the 61 to 95 amino acid sequences.
So is it conserved across the species as per your result?
Sir, please repeat the question.
Is it conserved across the species as per your result?
Yes, sir. The mammals have more or less conserved regions, because this is one of the most important protein, because it is related to our neurons. So it is very much conserved. Even you can see from the second graph, you can see there are almost 100% means 100% means uh, no or very minor, you can say even minor or no changes in that, in the sequence, which are related to mammals. Like here you can see in homo sapiens related portions, you can see gorillas, samaras, macaca. These are all almost, monkey or apes or that kind of old monkeys or new world monkeys and apes, gorilla. These have no changes. Even the faraway mammals like cats, dogs, or tigers and leopards, they also show dogs, cats, these also show very less means changes or no changes.
One
So
final question.
evolution shows that this is a very important protein, and the end region
Prabha,
has
Prabha,
been.
Prabha, we got the answers.
One final
Sir has
question is,
another question.
Yes, sir. Yes,
do you
sir.
think all these species what you have shown here get the disease?
Sir, I think this disease is mainly seen in means we can know about them only in humans. So Parkinson's,
It is conserved, then it should be, isn't it?
yes. But the importance means this species and the this study mainly shows that this is a very important protein and has been conserved from the very beginning of the of the classification of this taxa of mammal taxa. I wanted to show that only.
You.
This is a very important protein. Actually, this study also
Thank you, Prabha.
correlates.
It's a good work. Thank you.
Thank you, sir. Thank you so much. Should I leave the meeting?
Yes, please. Rashmi Sharma. Are you
Yes,
there?
sir. Am I audible?
Yes, please share your poster and quickly
sir.
introduce your topic and go to the result section and describe it.
Okay, sir. Good afternoon, sir. I am Rashmi Sharma from Department of Biotechnology and IT Jalandhar. I have performed my work on the topic formulation, characterization, and application of solid lipid nanoparticle. Solid lipid nanoparticles have gained significant attention as promising carriers for controlled drug delivery due to their biocompatibility and ability to enhance drug stability. Here, I have prepared solid lipid nanoparticle by using cocoa butter as lipid and lecithin as surfactant. Firstly, cocoa butter was heated at 40 degrees Celsius, and into it, I then added cocoa butter was heated at 40 degrees Celsius, and lecithin was also dissolved in distilled water, and then it was mixed into lipid phase. It was then stirred on magnetic stirrer for around 5 minutes at 400 rpm. Then it was sonicated, and after sonication, it was again stirred to form a uniform dispersion of nanoparticle. After preparation of blank nanoparticle, I have incorporated ibuprofen drug into it. In the corporation of drug, the drug was added to the lipid only when it was heated at 40 degrees Celsius. So it was an in situ addition of drug. And my results are, these are the results. SAM images of prepared SLN. Prepared SLN, SAM image of drug-loaded SLN. These are the FTIR spectra of lecithin, ibuprofen, solid lipid nanoparticle, solid liquid nanoparticle with ibuprofen drug, and then the XRD spectra of solid lipid nanoparticles, which I have prepared, and then the standard curve of ibuprofen, and then I have done the drug release study of ibuprofen, and drug release study was done by mixing 1 ml of SLN loaded with drug into 4 ml of phosphate buffer. saline. And then the application part is that this ibuprofen loaded SLN would be incorporated into a chitosan beta glycerophosphate thermosensitive gel to achieve localized and anti-inflammatory and wound healing effects. Thank you. Any questions, sir?
Thank you. I have simple two questions. Why did you use cocoa and why did you use ibuprofen? Is there any specific reasoning?
So cocoa I use because it was easily available in the market at a cheap rate, and it consists of three triglycerides, oleic acid, palmitic acid, and stearic acid. And these studies on these were conducted separately, but not any study or in combination of any of these two, but not all three. So I have selected this, and for ibuprofen I use this because it is easily available in market and it can be used and use drug for anti-inflammatory action, but it get released easily when you incorporate directly, but when you use it directly for consumption, but when it will be incorporated into solid lipid nanoparticles, then it will be easily it will be released, sustained release will be there, and you can control its release at the site.
So, but then that simply means it is not universally applicable, right?
Sir, it's universally applicable. You can use that SLN for containing drug and incorporate into a gel, and that gel when thermoresponsive gel, and you can inject that gel at the site of wound. That gel when goes into your body, it will be thermoresponsive, so it will be when it will be at room temperature, it will be at liquid, but when it goes into your body at that inflammatory temperature, it will solidify and will release drugs slowly at the site of wound. So the release, so the wound will get treated, but with sustained release. So longer effect of drug can be seen.
Thank you.
Thank you, sir.
Thank you to all the participants. Now we'll start with SOs within 5 minutes. Please, everyone be prepared.
पांच ही स्लाइड इनको रखना है ना। हाँ नेगेटिव मार्क हमें 5 स्लाइड होनी चाहिए।
If it exceeds 5 slides or there or 3 minutes presentation, there will be negative marking of 20.
Yeah, exceeds 5 slide. Point to yeah. उसका टाइटल दे दो। हाँ।
टाइटल स्लाइड इंक्लूडेड है क्या उसमें? टाइटल नहीं, टाइटल स्लाइड।
यहाँ पे 5 लिखा हुआ है। हाँ
Everyone, please attend your roll calls. Anushri Pant, Smriti Singh, Vanchika Garg, Ashwini Kumar Verma, Shania, sorry. Do we have Shania? Okay. Shubhada Shankar Chaudhary. Okay. Diya Singhal, Rekha Kumari. Okay. We do. Naveen sir is also not. Vanchika Garg and Shania. Plus, Mr. Naveen Kumar is also not coming.
Yeah, he will come with it. Naveen Kumar then? कम हो गया।
I'll clear the timings to all of you. Your time limits would be 10 minutes. First tab will be at 5 minutes and the second tab will be at 7 minutes. Rest 3 minutes will be for your questions.
So, Anushri Pant, first candidate.
Afternoon to everyone present here. I am Anushri Pant, research scholar from Department of Biotechnology, Dr. B.R. Ambedkar National Institute of Technology, currently pursuing PhD. I'm audible to everyone here. Okay. Currently pursuing PhD. Currently pursuing PhD under the guidance of Professor A.K. Jana. The topic of my work is biochemical characterization and molecular evaluation of lipases for eco-friendly detergent formulations. So what are the problems that are associated with synthetic detergent? That they contain surfactant, bleaching agents, phosphates, and many artificial agents, which when discharged into the wastewater, they cause environmental problems. So environment-based detergent are an alternative to such kind of detergent. And lipases is one of the enzymes that can be used in detergent formulation, and it is increasing at a compound annual growth rate of 4.2%. So now the scope of work is that most of the existing studies in the work has been done in purified enzyme, which are not cost effective. And the work that has been done has been evaluated on the basis of simple titration method or visual assessment. Moreover, more mechanistic work need to be done. And the quantification of enzyme activity has not been explored. So a systematic study on the quantity of enzyme need to be explored. This is the rationale of the study in which lipase has been isolated. After that, enzymatic characterization of the enzyme was done. It is commercial with the help of commercial detergent. It is tested, and after that, it worked as an detergent additive, and lipase and substrate interaction was tested for the stain removal assessment. The purification of lipase has been done in which the specific activity increases with that of protein content decreases. This is the SDS-based analysis which revealed the molecular weight of approximately 63 kilodalton of lipase enzymes. The physiochemical characterization of enzyme revealed that the enzyme is stable at 40 degrees Celsius at an alkaline pH of 9. It is also stable in the presence of various metal ions, anionic and ionic surfactants, as well as inhibitors. This is the visual assessment of the stain removal in which 3 stains are taken and the enzymes. As we can see that lipase is when incorporated with that of 1 gram per liter of detergent, it gives effective result to that of 10 grams per liter of the detergent. So we can conclude that if we incorporate enzyme with that of low detergent concentration, the detergent amount will be reduced by 10-fold. In this way, the detergent reduction is there. The wash water analysis was done with that of titration method and FTR analysis, which revealed that the ester peak decreases with the increase in hydroxyl peak, which result in the breakdown of triacyl glycerol into monoacyl and diacyl glycerol. The free fatty analysis revealed that the amount of free fatty acid released after treatment increased. This is the molecular docking assessment in which the enzyme from the asparagine lipase is docked with that of 3 substrate, that is trialine, linoleic and resinolic, and the more the negative binding energy, it means the higher the binding interaction. And after that, the 2D visualization and 3D visualization of enzyme substrate is done. So from the work, it can be concluded that enzyme is an alternative for the enzyme for the detergent formulation, and the use of detergent can be reduced if we incorporate any of the enzyme. And the partially and crude enzyme, when incorporated with that of detergent, it will be more cost-effective process for the enzyme for the detergent formulations. Thank you.
So, simple question here is,
yes,
lipase is known, right? Why
yes,
do you need to characterize it?
Because it is isolated enzyme. I have isolated that enzyme microorganism. After that, I produced lipases, and that need to be characterized.
Did you express it in something? Or
no, this is the no. It is the isolated, and then after I have characterized.
Isolation, how much did you get out of it?
I have 53 isolates, from which I have first identified that on the basis of qualitative analysis, that is rhodamine B and tributylin. After that, lipase assessment was done, and on the basis of highest lipase production in MSM media, which is supplemented with yeast extract and olive oil, this strain is highly produced and it is alkaline strain, so it is much suitable for detergent additive applications.
Right. So how much it is? Like how many mg per liter? Or
yeah, I have mentioned this. The crude enzyme has approximately of 126 unit per ml of enzyme, and after partially purification, the enzyme activity increases.
Okay.
Yeah.
It increases.
Yeah, increases because there is.
How it is going to be cheaper? That is what you are claiming, right?
Yeah.
So how it is cheaper? Because this whole process involves a lot. It is not really very high unit per ml.
No, according to the literature.
Do you need to add in your detergent? How many units?
I have added 5 ml of enzyme, which approximately 1 unit contains 204 unit of enzyme. So for 5 ml, we can calculate 1, it. Yeah, approximately 1000 unit. For 1 grams per liter of detergent.
Okay, and how much it will cost?
Sir, actually cost analysis, I'm going through this, and after that,
It's not cheaper.
sir, due to the partial purification of the enzyme, it is cheaper. But if we purify the enzyme that is already available in the detergent formulations, as I have already worked with the detergents that is already present in the market, enzyme-based detergent that are approximately cost 500 to 600 rupees per liter. So if we incorporate that in the enzyme, that are purified lipases which are incorporated in that detergent. If we will use that enzyme which is not purified, which is partially purified, the amount of the cost will be reduced. And I'm working on that cross prediction.
So how much cost right now it is?
Actually, I have not calculated the cost, but approximate.
No, I'm emphasizing on that because this is the theme of your topic.
Yeah,
right? You are
yes.
reducing the cost.
Sir, my next work is on cost analysis.
Okay.
Yes. Yes, ma'am. Sure.
Yeah, I also have the same question.
Ma'am, Nova enzyme has approximately, Candida rucosa I don't know that is from Nova enzyme or not. Candida rucosa is of sigma lipase that have 700 unit per mg of enzyme. So I have worked with that also. Ma'am, yes, ma'am. I am working on that, the calculation of that enzyme with that of Nova enzyme or comparative analysis of that enzyme. I have used synthetic detergents as well as enzyme-based detergent that are already present in the market. This is the comparison, and this is the enzyme-based detergent which I have used, and that are the synthetic detergent.
Check the stability of.
Yeah, I have checked the stability of the enzyme, and I have also checked the lipase activity, which is all, which is the.
Not shown the result.
Yeah, I have not because it is the time constraint and only 10 slides I have to show. This is what washing has been done in normal tap water in alkaline. The pH what is present in the tap water, normal pH. No, alkaline, neutral pH, water pH, normal tap water. Slightly alkaline. Acidic, sorry. Yeah, it is working on that, but it is not that much stable. So I will just incorporate in that, and optimization later on. Optimization will also done on the detergent quantification as well as on the enzyme quantification. Yes, ma'am.
If you not further purified it, you just mentioned partially purified.
Yeah, no, sir. Actually, my main motive is to work on the cost-effective lipases. So that's why I have not purified that lipases.
Okay.
Yeah, I am doing that work and I am purifying it. Yes, sir. Yes. Okay, sir. Okay, thanks. Thank you.
Next, Smriti Singh from Banastili Vidyapur.
Very good morning, everyone. Myself, Smriti. I am research scholar at Biotechnology Department of Bioscience and Biotechnology. The topic of my presentation is in silico target prediction and molecular docking from Caligonium chromosome flower buds metabolite reveal potential protection against fluoride-mediated liver injury. So nowadays, the drug discovery, the computational biology has a huge role in drug discovery. The plant Caligonium chromosome is commonly known as the ARTA. The plant Caligonium chromosome is commonly known as ARTA, has high medicinal property and contains antioxidant, antimicrobial, anti-inflammatory, etc. activities. So it integrates two steps. One is the traditional method of extraction, which include hydroethanolic fractionation and modern computational biology, in which the phytocompounds analyzed through GCMS was then evaluated through molecular docking with hepatoprotective targeted proteins against fluoride-induced hepatotoxicity. So there are the three fractions, chloroform, ethyl acetate, and butanol. The ethyl acetate fraction shows high flavonoid phenolic content and high pyrrhic-reducing antioxidant power. Through GCMS, phytocompound analysis was done. The 4 compounds were selected on the basis of their hepatoprotective activity, and further studies were done with these compounds. The data represents molecular docking of these 4 phytocompounds with caspase and TNF-alpha. From these 4, phenol 2,4-bis 1,1-dimethyl ethyl shows the lowest binding affinity with caspase and TNF-alpha. This is the egg plot of the 4 compounds, which shows its GI absorption and blood-brain barrier. So the 4 compounds follows the Lipinski rule, drug likeness, and it has the water solubility and the GI absorption and permeability. All 4 compounds it's up to the so the phenol 2,4-bis 1,1-dimethyl ether shows low binding energy with TNF-alpha as compared to the caspase 3. And the conclusion is that the phytocompounds may contribute to the anti-inflammatory and apoptosis modulated effect and strong inhibition toward TNF-alpha. And the significance of my work is this finding supports my hypothesis that Caligonium chromosome flower bud fractions molecules with reasonable phytokinetics behavior. Thank you.
You're in which semester of your PhD? Second year. So are you planning to do MD simulation also?
Yes, sir. Yeah, further.
Do you understand what is MD simulation? Because there is lots of oxidative stress activity in the liver, for which you are designing this, yeah, right? So what is MD simulation? Why MD simulation is important? And also talk about more about the ADME analysis that you have done. Because these 2 things are very important in case of liver.
Sir, actually, so in vivo activity.
In vivo नहीं पूछ रहा हूँ. हाँ, ठीक है. हाँ, तो अभी एकदम probably हाँ वही. Next is Ashwani Kumar Verma from IIT Guwahati.
Good evening, everyone. I am Ashwani Kumar Verma. I'm a PhD student at IIT Guwahati under the supervision of Professor Arun Goyal. So the topic of my presentation is functional characterization of a obligate cytobiohydrolase from acetyl of level. So as we all know, sustainability is the essence of the near green future. When we compare the relative advancement that have been achieved in case of the other types of the biowaste, for example, lignocellulosic biomass, very little has been achieved in case of the marine crustacean biomass. Marine crustacean biomass is being generated in abundance from various industrial as well as domestic processing units that work exclusively with shrimps, crabs, lobster, krill, etc. So despite beholding immense biotechnological potential in various fields of agriculture, food, pharmaceutical, and medicine, these wastes are generally discarded into the nature, highlighting a serious concern for environmental pollution, but also representing a serious resource waste. So chitin, it is an important component of such marine crustacean waste. It constitutes about 15 to 45% of the total crustacean waste. It is a linear homopolysaccharide consisting of N-acetyl glucosamine linked together with the help of beta-1,4 linkage. So chitin depolymerization, which can be achieved with the help of biological as well as chemical approaches, where biological approaches involve the utilization of a group of enzymes which are referred to as chitinolytic enzymes. And these are much more specific as well as cost effective compared to the chemical approaches which involve the use of HCl as well as NaOH. So in this project, we first conducted the in silico based genome mining of an organism, which is Acinetobacter mellifera. It is an anaerobic microorganism which was first isolated from an anaerobic slab. It is well studied for its cellulolytic as well as hemicellulolytic ability, but its chitinolytic potential is yet to be explored. So after genome mining, we identify a single gene which was encoding for GH18 chitinase. The gene which contained a single signal peptide, a single catalytic domain as well as a toxin. So we PCR amplified just the catalytic domain and left the rest of it. So the amplified gene was then flowed into PET28T vector and was overexpressed under the induction with 1 millimolar IPT. The recombinant protein was subjected to NIMTA purification, in which we were able to obtain a single-step purification of this protein as shown in figure 11. Next, we wanted to check what is the native state of this protein, for which we conducted native phase analysis, in which we observed that the native state of the protein is a dimeric state. Next, since we are establishing this protein for industrial sector, we wanted what is the stability or solubility of the protein at high concentration, for which we conducted the zeta potential analysis, in which we obtained that at concentration approaching 4 mg per ml, it had high negative potential of around minus 57 millivolt, indicating that it has high protein-protein repulsion at high concentration and high stability. Next, we wanted to check what is the activity of this enzyme on variety of the raw as well as the chemically modified chitinase substrate, in which the enzyme showed high activity of 27 and 26 unit per mg against carboxymethyl chitin from shrimp and carboxymethyl chitin from crab. All of these chemically modified substrates were prepared by us only in the lab. Next, we checked the temperature optima of the enzyme, in which the enzyme showed activity at a wide range of temperature from 20 to 100, where the maximum activity was obtained at 60 degrees. For melting temperature analysis of the protein, we checked what was the unfolding of the protein when the temperature increased. That is, we checked the hydrodynamic diameter of the protein, where we observed that there was a drastic increase in the hydrodynamic diameter when the temperature increased to 63. We also checked the pH optima as well as the pH stability of the protein, where the enzyme showed highest activity at pH of 6.5 in the sodium phosphate buffer 50 millimolar concentration, and it retained more than 60% of its activity at pH range of 3 to 8, indicating that it is a highly thermostable as well as a pH stable enzyme, making it relevant for industrial applications. Next, we wanted to check what is the product the enzyme produces when it is acting upon the variety of the chitinase substrate, for which we conducted the thin layer chromatography in a time-dependent manner. When the enzyme is acting upon the colloidal chitin, we observe that starting from 5 minutes till 12 hours, there was a production of only one type of substrate, which was N-diacetyl chitobiose. and the total amount of the total reducing sugar was reaching up to 0.5 mg per ml before reaching saturation. Similarly, when the enzyme was acting upon the carboxymethyl chitin from shrimp, only a single type of the product was produced starting from 5 minutes till 12 hours, and the maximum TRS was almost similar, that is 0.5 mg per ml. Similar results were obtained for crab colloidal chitin, that is same chitobios was formed starting from 5 minutes till 12 hours, and the total TRS was 0.5 mg per ml after 12 hours saturation, 12-hour incubation before attaining saturation. Next, in order to check the integrity of the end products, we used multi-top analysis in which we observed that there was a single predominant peak at 447 as well as 463, which correspond to the potassium as well as the sodium adducts of the chitobios, indicating that it is a pure obligate type of the chitobios, chitobiohydrolase, which is a novel type of hydrolase since it is not reported in literature previously. Since it is a exotype of the enzyme, so we wanted to check the processability of the enzyme, that is capability to remove the successive chitobio from the same polymer, in which it showed high processability index of around 11 and 10 for carboxymethyl chitin from shrimp and carboxymethyl chitin from crab. Next, we wanted to check what is the application of this enzyme in a real life work, in which we collected the raw waste shrimp shells from the local market. We washed it with water in order to remove all the flesh and dirt particles. Then we dried it and physically pre-treated with grinding. Since chitinous waste contains calcium carbonate, so we needed to treat it with hydrochloric acid, 2% HCl. After which it was washed with water in order to obtain the neutral pH. 100 mg per ml of concentration of enzyme was used for the pre-treatment of pre-treated biomass. After which the product formation was analyzed using the TLC, in which we obtained that the enzyme was producing chitobios as previously seen, and the total TRS was about 0.35 mg per ml. So the future work, which includes the synergistic action of this enzyme with N-acetyl hexosaminidase, as well as with optimization of all the pre-treatment parameters. Finally, I would like to thank my supervisor as well as LPT conference for giving me a platform to represent my work.
Okay. So first thing, how did you know that it is a dimer? You said this enzyme is in dimer when you showed the SDS page.
Yeah, actually, I have not shown the native page revealed that since we use the 10% TS as a standard, we are not confirmed that it is. We cannot say just by native page. We have sent it for SAX analysis, that is small angle restless scattering. So after that only we will be able to say that it is a dimeric state. So just we observe that the band, we observe the clear band above 66 kilodalton. So I was predicting since the theoretical molecular weight of the protein is about 43 kilodalton. So it was above 66. So I was assuming that it must be.
That's the point, because you didn't show anything validating that it is just fair enough. Okay. So then at the end, you also mentioned that real-time benefit, but you didn't show me any real-time benefit. You cleaned, you got the enzyme. Yeah. So what next?
Sir, actually, this work is still under study. Actually, the main application of this enzyme, I will be using my chitobios, and I will be immersing it into bacterial cellulose to produce a wound healing material. So currently, I will be optimizing to obtain a maximal TRS from raw shrimp waste. Then the TRS that is I obtain, that is chitobios, it will be immersed in bacterial cellulose. And the material that I obtain will be used I will analyze for the ideal parameters of a wound healing material, for example, absorption, antibacterial, antioxidant. If it qualifies for all of it, then I will be able to say it.
Okay. Thank you.
Next, Shubhada Shankar Chaudhary from from IIT Guwahati.
Good evening, everyone. Myself, Shubhita Chaudhary. I'm an MTech student from IIT Guwahati under supervision of Professor Arun Goyal. So my title of today's presentation is Cloning Expression and Purification of N-acetyl beta-dehydrosaminidase belonging to glycoside hydrolase family 20 from bacterial oil. So the major motivation of my research is the crustacean waste. The extreme extreme expansion of industry shows the extreme expansion of industry shows the environmental pollution which
आराम से घबराना नहीं है, ठीक है? 10 मिनट है आपके पास, आराम से बोलो, we will listen.
So the vast industries are producing so in industries they are producing the crustacean waste in vast quantity which results into environmental pollution. So here the main focus is the chitin, which chitin which makes 5 to 6 million tons of chitin was discarded into the oceans, which contribute into environmental pollution. So here is the structure of chitin, which composed of repeating units of N-acetyl glucosamine linked to the beta-1,4 glycosidic bond, glycosidic linkages, which are strongly hydrogen, which are composed of strongly hydrogen bond, which makes it highly crystalline. So the tight packing of the chitin shows the insolubility in water. So here the main organism which we selected is Bacteroides ovitus, which was initially isolated from the gut of the cattle, and the enzyme it consists of 4 types. In type 1, it contains the catalytic domain. In type 2, it shows the catalytic domain along with the GH20B domain at N terminal. In type 3, it shows the catalytic domain along with the GH20C domain. And in type 4, it shows the catalytic domain along with GH20B at N terminal and GH20C at C terminal. So for selecting the molecular structure, we did some techniques which is NCBI blast analysis. From that, it shows the highest similarity of 29.771% with sericium lustrations. And for confirming the molecular structure, we also designed the model with the help of AlphaFold 3, which shows the 3 domains, one domain of beta sandwich model at N terminus. It shows the QT2 CTM. But second, here is the mixed alpha beta shades domain, which confirms the GH20P domain. And here is the alpha beta team barrel like structure, which shows the So after that, for cloning techniques, we perform the PCR amplification, which shows we considered different annealing temperatures, which was 58, 60, and 62, and the intense band was observed at 60 at temperature 60 degrees Celsius at desired band at 1.3 kb. After that, we isolated plasmid and we did the restriction digestion with the help of with NHE1 and EcoR1, and which shows the desired band of plasmid at 5.369 kb and of gene at 1.303 kb. So for confirming that clone, we did the transformation we did transformation in E. coli BH5 alpha cells and select the colonies, and after select the colonies, and from those colonies, colony number 3 shows the desired band at 1.303 kb. And for further confirmation of clone, we also perform the plasmid PCR and double restriction digestion, which in which it also shows the desired band at desired size, which is 1.303 kb. Here the conclusion was the clone was confirmed by clone was confirmed. So after that, we express it in the Rosetta cells and select the colonies for after transformation. We select the colony and we express. So the after expression, the desired band shows at the desired size of protein, which was 49 kilodalton, and from those we did the purification. We did the purification of at purification. We ran the SPS gel and did the analysis, and the desired size was, and the band was observed at the desired size, it was 49 kilodalton. So after that, we also did the native gel, run the native gel for showing the for observing its dynamic state, and as it is shown, the protein is 49 kilodalton. After analysis of native phase, it was observed, single band was observed at 98 kilodalton, which also approximates. So we can assume that it is in dynamic state. For confirmation, we also did the DLS, which is dynamic light scattering. We did at the different concentration 0.3, 0.5, 0.8, and 1 mg per ml. So in this, we also observed that it also shows the hydrodynamic diameter approx 87.3 nanometer, which indicates its protein in dimeric state. So after that, we confirming its solubility of purified protein GH20, we did the zeta potential. So it shows the highest negative potential, which is minus 38.79 millivolt at concentration of 0.8. So it also indicates that it is highly soluble of purified protein. So overall conclusion, it's that we.
आपने सारा काम खुद किया है?
Yes, sir.
ओके, कुछ पूछूं मैं? सीक्वेंसिंग कराया था उस प्लाज्मिड का?
Sequencing is 3D sequencing also, and
ये आपने बताया नहीं सीक्वेंसिंग कराया? ओके, रोजेटा सेल्स में क्यों एक्सप्रेस किया? कोई खास कारण?
We did in codon plus also, and the band was observed in Rosetta after Rosetta.
तो कुछ पता है आपको रोजेटा सेल्स क्या है?
It helps in expression.
Helps in expression, but do you know something unique about it? Because usually Rosetta में नहीं करते हम लोग.
We perform in codon plus BL21 and Rosetta also.
Okay, so you did it in BL21 and you did it in Rosetta. So that's exactly what I am asking. What is the difference then compared to BL21 and Rosetta?
Proper results in Rosetta.
Right. ये सब आपने खुद किया है?
Yes, actually, I did my work under the supervision of my PhD and Arun Professor.
नहीं.
Yeah.
कितनी डिग्री पर एक्सप्रेस किया इसको आपने? प्रोटीन कितनी डिग्री पे एक्सप्रेस हुआ आपका? आईपीटीजी से इंड्यूस किया होगा। एक टेम्परेचर क्या रखा था? 24 श्योर? एसडीएस जेल की बात हो गई। अभी आपने प्रोटीन इंड्यूस किया, आईपीटीजी डाला।
आईपीटीजी 24 डिग्री से
24 डिग्री
आफ्टर इंडक्शन
कितनी देर के लिए रखा? 1 आवर के लिए, 2 आवर ओवरनाइट रखा?
overnight after induction we keep it for overnight and then after that we
पूरा अंदर जैसे तो ब्रेक कैसे किया? प्रोटीन कैसे निकाला?
with the NINC column we purified our protein.
You must have broken the bacteria first, isn't it? तो क्या होगा ना, निकल एनपीएच तो बाद में आएगा। Did you break the bacteria?
I didn't get it.
So you induced.
Yeah.
What did you do after that?
We induced. After that we select from culture.
You centrifuged.
Yeah. We
centrifuged.
Then from pellet we run in SDL.
No. So you are missing something. That's what I need to know.
We induce and after that we collect from that culture.
Did you break it? Pellet to break it?
Yeah, we centrifuge it.
Centrifuge it. You pellet. उसके बाद निकलेगा कैसे उसमें से प्रोटीन?
Actually we do sonigation.
You did the sonigation. Very good. So you did the sonigation. कैसे किया? कितना रखा? इसी के मैं पूछ रहा हूँ। आप आगे मास्टर्स स्टूडेंट हैं। I need to know कि you did it.
Sonigation for 30 to 45 minutes.
45 मिनट इतने में तो सारा प्रोटीन बर्बाद हो जाएगा।
It gets 30-45 if the ice is melting or what, but it is around 30 minutes. Sonigation is done.
30 minutes.
Yeah.
लगातार?
No, we set the amplitude also. So 10 for 10 seconds.
Okay. किस चीज में किया? कितना था आपका कल्चर?
For 400.
400 ml. तो आपने फाल्कन में किया होगा फिर? In beaker, glass beaker में. चलो, thank you. Good job.
Next is Divya Singh.
A very cheerful good afternoon, everyone. I am Dia Singhal, currently pursuing BSc Forensic Science from Amity University, Noida. Today, I will be presenting my review work on nanoparticles-based colorimetric sensor for rapid detection of novel pesticides. This study focuses on how nanotechnology can help us detect pesticide residues cost-effectively, rapidly, and even on site. Today, I will be discussing about the basic introduction, objective of the study, literature review, methodology, performance evaluation, challenges and limitations, along with conclusion with future outlook. Let's start with the basic introduction. Pesticides are essential in agriculture, but their residues remain in water, soil, and food systems. But the residues remain in water, soil, and food system. Their accumulation cause harmful effects on health and environments. Several analytical techniques are HPLC and GCMS are used, but they were accurate, but they're time-consuming, costly, and not suitable for on-site monitoring. This review evaluates the sensing performance and limitations of different nanocomposites to identify suitable platforms for real-time monitoring and field-based pesticides. Now, the main objective of this study is to investigate different nanoparticles for colorimetric sensing of novel pesticides. In this study, I evaluate major nanoparticles such as gold, silver, carbon quantum dots, graphene-based, and zinc oxide. And these were evaluated based on their sensitivity, LOD. LOD stands for limit of detection. It is the smallest amount we can detect, response time, and field applicability. By comparing all these, moving on, I would like to tell you about my literature review. Recent studies highlight the growing application of nanoparticle-based colorimetric sensor for rapid pesticide detection in environment and food samples. So I analyzed major nanoparticles, which I have mentioned. Let's start with gold nanoparticles. Gold nanoparticles exhibit LSPR. LSPR stands for Localized Surface Plasmon Resonance. LSPR stands for Localized Surface Plasmon Resonance. They are a collective oscillations of electrons present on the platform of gold nanoparticles. When light falls onto them, they changes their color. They are highly sensitive. Silver nanoparticles provide optical properties and high surface reactivity. Carbon nanotubes and graphene oxide improve adsorption and sensing efficiency. Zinc oxide nanoparticles provide chemical stability and selectivity. Although I have mentioned analytical techniques were also used, but analytical techniques are time-consuming, costly, and not suitable for on-time monitoring. I have mentioned analytical technique. Those were used for HPLC and GC. HPLC stands for High Performance Liquid Chromatography. It's a laboratory analytical technique that is used to separate, identify, and quantify compounds based on their high pressure, compound based on high pressure, under high pressure. GCMS stands for Gas Chromatography and Mass Spectrometry. It's a separation technique that separates compounds based on their mass to charge ratio. Therefore, nanoparticle-based colorimetric sensing platforms are emerging as cost-effective, portable, and real-time alternative for pesticide detection. This is a systematic review of nanoparticle-based colorimetric pesticide sensors. Now, the performance was evaluated based on their sensitivity, selectivity, LOD, and response time. Nanoparticle-based sensor demonstrate detection limits in nanomolar to picomolar ranges, which is extremely sensitive. These were comparative analysis of nanoparticles by HPLC and GC, and they are found to be rapid, simple, and portable techniques. Now, as nanoparticles techniques are rapid, portable, and simple, here are a few challenges also. While nanoparticle stability can be affected by pH, temperature, and storage, which can reduce their shelf life. Limited selectivity for structurally similar pesticides compounds, need for standardized sensing protocols and real-time mechanisms. Last but not least, I would like to conclude my presentation by nanoparticle-based colorimetric sensors provide a rapid, visually simple, and cost-effective platform for pesticide detection. Therefore, analyze gold. Gold is considered the most suitable nanoparticle because it is highly sensitive and it uses LSPR. Silver is also considered a very nice nanoparticle for this. Carbon and metal oxide based nanoparticles enhance stability and practically usability. Selective surface functionalization enables nanomolar to picomolar detection, which is extremely sensitive across multiple pesticide classes. Future focus should be based on improved performance in complex matrices, standardized validations protocols, and integration with smartphones-based monitoring systems. Thank you.
So are you planning to make something?
Yeah, sir, this is my review right now. I'm looking forward to do research further on my master's in this topic.
Good. All the best.
Thanks.
So next is Rekha Kumari from Vanasthali Vidyapeeth. Tap it.
Good evening, everyone. Good evening, everyone. My name is Rekha Kumari from Banasli Vidyapeeth. I am a research scholar, and today my presentation is on bioactive profiling and computational evaluation of catfish epidermal mucus as a natural source for dermatological formulation. For this, first of all, we have collected the mucus from the fish, then we stored it, and then further we analyzed the phytocompounds which are present in mucus through GCMS. Then further evaluation, we use computational tools, and then we analyze the results for further future significance. Now, why we choose fish mucus as this proposal? Because it contains some important innate immune responses, which includes protease, AMPs, and lysozymes. It also contains gelatin. It also contains some important proteases enzymes. And as I mentioned over there, it also contains some AMPs that are used as antimicrobial activities. The study includes mainly 3 objectives. First of all, the collection of mucus from the fish. Second one is the analysis the phytocompounds from the mucus, and then evaluation the dermatological potential of the components present in the mucus. Next, similarly, we collected a mucus from the fish dorsolaterally side. The fish length was 19 to 25 nearly centimeter, and weight was 50 to 55 centimeter. And then we sterilized, we centrifuged it with equal amount of physiological saline. Then we prepared the sample for the GCMS and FTIR analysis. Then the result we get from the GCMS. Then we used computational tool to analyze the results. Next. This some main compounds we found out from the GCMS. These are compounds which have mainly the antioxidant activity, antimicrobial activity, and some having anti-cancer or hepatoprotective activity also there. Then we run for the FTIR analysis. By FTIR, we know about some functional groups which may lead to help us to find out which biomolecules present in our sample. Then in silico tools, we use pass online. Through pass online, we know the probability activity of the subproteins regarding anti-inflammatory, antimicrobial, and probability of activity and probability of inactivity. The probability of activity is nearly or more than 0.7. Then it is good. Having good this type of activities like anti-inflammatory, antimicrobial, anti-diabetic activity. Most of the compounds we selected here have a higher value more than 0.7 of PA. That is activity probability. Next, we perform Swiss ADME and PKCSM. Through this, we analyze that most of compound having log p-value is in between 2.7 to 3.7. That is may be useful for topical formulation. And next, some through PKCSM, we see that skin creamage value are also acceptable here, and surface area of this compound absorption is also high, and it also reveals the skin sensitization is also in positive way. Yes, it is maybe in positive way or negative way. It is further be analyzed by the in vivo experiment. And I conclude this may be a good proposal for further cosmetic industry for drug delivery, and it may be a good work for further development. These are the references. Thank you.
What is your basic aim, like what to achieve in your PhD work?
Sir, areas PhD of my area is different. PhD, हाँ जी sir. This is our additional work we want to perform with fish mucus.
आपने फिश को खुद होल्ड करके म्यूकस कलेक्ट किया?
Yes sir.
कितना बड़ा था आपका फिश?
सर मेरा था.
आपने क्या लिखा है वहाँ पता है?
पहली साइड में
हाँ तो वो
50
kg लिखा था जो
50 kg
16 या 17
50 kg का फिश था 19 सेंटीमीटर का। उसको तो आप उठा ही नहीं पाएंगे।
पहले है ना कुछ प्रेजेंटेशन दे रहे।
और कुछ
कलेक्टिवली लिया था।
तो कैटफिश क्यों लिया? कैटफिश तो डेंजरस होता है, आपको पता है?
Why sir?
आपको नहीं पता?
स्टिंगिंग फिश है इसलिए यार ओमनिवोरस है किस कारण से?
वो आप देख लीजिएगा।
नहीं सर,
I'm wondering why did you choose catfish? It's not that.
So is it reported somewhere? Did you have any reference paper where it is being reported that it could be a problem? Yeah.
हाँ सर, जैसे कि हमारे इसमें सर ये है स्नेल कंट्रोल कॉस्मेटिक्स के लिए बहुत ज़्यादा
स्नेल अच्छा है ना?
हाँ स्नेल अच्छा है। तो सर इसका भी म्यूकस जब हमने वो किया ना सर, कई सारी से जेब्रा फिश का कर रखा है सर, बहुत सारी
जेब्रा फिश तो इतना छोटा।
हाँ तो सर उसका भी कर रखा है सर, एपिडर्मल सेल्स लेकर के फिर उसका उन्होंने फर्दर कर रखा है सर। तो ये एक इनिशिएटिव है सर, ये फेल भी हो सकता है, ये सक्सेस भी हो सकता है।
So what type of dermatological aspects it will target?
सर जैसे कि सर अब मैंने सर जैसे डॉकिंग किया था।
हाँ what type of dermatological
जैसे सर स्किन ब्राइटनिंग में भी हो सकता है सर ये, और सर जैसे कि इसके अंदर 70 to 80% वाटर है।
हाँ तो वो आगे डॉकिंग करोगे, आगे काम करोगे, वो सब हाँ वो तो कर रखा है वो अपने।
हाँ तो सर उसमें सर जैसे MMP9 है और दूसरा हमारा है कोलाजिनेस है, ये सब क्या करते हैं सब मैट्रिक्स में कोलेजन को डिक्रीज करते हैं। तो हाँ एजिंग कोज करते हैं। तो अब इसके साथ बाइंडिंग है, इसने बाइंडिंग ने सिर्फ ये बताया कि बाइंडिंग नेगेटिव है, मतलब कि तो अच्छा रिजल्ट हो सकता है, पर वो आगे जाके पॉजिटिव वे में भी हो सकता है। और नेगेटिव में भी उसका वैलिडेशन नहीं हुआ है सर। अब एक आइडिया है और हम ये एग्जीक्यूट करना चाहते हैं।
It confirms this is a that sound.
We are deeply honored to have our esteemed chairs guiding the session with their expertise, insight, and academic excellence. Now, it's my privilege to invite Dr. Subhash Nigam to the stage to honor and facilitate our respected chairs for their valuable presence and guidance. Thank you, everyone. This was a very exciting session. Thank you all for participating. Now, the next session will begin in 10 minutes.
Thank you so much.
Thank you.
Thank you so much.
answer. So we have this chapter.
हाँ ये वाला तो बहुत अच्छे से हमारे पास वो आए भी नहीं ना
बच्चे नहीं आए ना तभी बच्चे टाइम हो जाएगा ना ये वाला ये तो हो जाएगा ये वाला काम कर देंगे लास्ट चलो कोई बात नहीं कोई बात नहीं नाम लिख दिया था तुमने हाँ जी मैं लिख दी अभी मत लिखो एक अजय चौहान का अजय चौहान डॉक्टर अजय चौहान
तुम्हारे डॉक्टर
आ रहे ये तुम अजय चौहान अजय चौहान
बोलना चाहता
ये दे दो यही काम करना था लैपटॉप यही गया सही कह रही है
We will start in just 5 minutes, and we are requested to start with Ananya Padhe first. Then we'll go with serial wise. Ananya, please be ready.
Okay, thank you so much. Is my voice audible?
Yes, your voice is audible. We'll start with you within 5 minutes.
Okay, thank you so much.
Good evening, sir.
गुड इवनिंग कैसे हो
Hi, Dr. Vivek. How are you?
हाय कृष्णा फाइन
We will start with Ananya Pandey, right?
We'll start with in 5 minutes.
Excuse me, sir.
Yes.
Actually, I have a small query that if I can record the session of my part and keep it with me, is it okay?
No, you have recording it, so no need of that, I think. But if
Actually,
if you are recording, that's fine. No problem.
I can record now because I have to show it to my guide.
Oh, okay, okay. You can record.
Thank you so much. I'll start in 2 minutes.
Okay, we'll inform you when you have to start. You can start recording then.
Okay, thank you so much.
Yes, Shivani, it is the online poster presentation.
Ananya, are you there?
This is introduce first. Yeah. Distinguished delegates, esteemed scientists, honorable chair, respected faculty members, and dear participants, good morning. Good afternoon. I am Navarati, and it is my privilege to welcome you all to the International Conference on Sustainable Biotechnology and Bioinnovation, SBB 2026, being held from February 25th to 27, 2026. This prestigious conference is organized and hosted by Amity University, Uttar Pradesh, in association with Biotech Research Society, India, International Bioprocessing Association, IFIB BIOP, and the Center for Energy, Environment and Sustainability. SBB 2026 brings together brilliant minds from academia, research, and industry to explore innovative solutions in sustainable biotechnology and bioinnovation. I am privileged to introduce the session chairs, Dr. Krishna Gautam and Dr. Ajay Chauhan. And online we have Dr. Rajkumar Regar. Dr. Rajkumar Regar, PhD, is a microbial biotechnology specialist with over 12 years of research experience in environmental genomics, antimicrobial resistance, metagenomics, and biodegradation. He currently serves as research associate at the Central Research Institute of Homeopathy and completed his PhD in microbiology from Babu Banarsi Das University with research at CSIR Indian Institute of Toxicology Research. His work spans microbial diversity, analysis of major Indian rivers, antibiotic resistant, gene profiling, microbial bioremediations of contaminated sites, and evaluation of homeopathic drugs using in vitro and in vivo models. He has He has authored 23 peer-reviewed publication combined IF greater than 150 and IH index 10, contributed genome sequences of NCBI, and secured participated in projects funded by ICMR, DBT, CSIR, and the Ministry of AYUSH. Technically, proficient in advanced metagenesis, molecular biology, high throughput sequencing bioinformatics tools, chromatology, mass spectrometry, and SEM. He has presented at national and international conferences, received conference award, and actively contributes to scientific research monitoring, mentoring, and interdisciplinary collaborations. At this point, it is my honor to formally hand over the proceedings to our chair. I invite you preside over the session, call upon the participants, and guide the presentations and evaluation. The stage is now yours to let the discussion engage with our presenters and shape the scientific disclosure of SBB 2026. I would like to clarify about the time limits. It's a poster session, so each and every delegate will get 3 minutes. First tab will be at 2 and 30 minutes, and the another tab will be at 3 minutes. Then the chair will be ask your question if they have any, and then you can leave. Thank you.
Hello. So first, I would like to call Sevani Tripathi to. Okay, Ananya Pade, first candidate who is going to present her work.
And so is my camera visible and my poster visible?
Yes, share your poster.
Is it visible?
Yeah, Ananya, make it slideshow, please.
Is it now visible?
Yes. You may proceed.
Good evening, respected chairpersons, distinguished delegates, and fellow participants. I am Ananya Pandey from the Amity Institute of Environmental Sciences, Amity University, Uttar Pradesh, Noida. I am presenting my research titled Zero Hunger, a Goal of Sustainable Future, Status and Implication. This research not only evaluates India's food security status, major challenges, and its implications for achieving the Sustainable Developmental Goal 2, Zero Hunger. Despite being a major agricultural producer, India continues to face serious issues of hunger, malnutrition, and large-scale food wastage, whereas around 195 million Indians remain undernourished, with women and children being most affected. At the same time, nearly 1/3 of the global food is wasted in India, whereas 79 million tons of food is wasted before the consumption. This contrast between hunger and waste highlights gaps in distribution, access, and management. Whereas in the COVID-19 lockdown, the exposed structural weaknesses in the food system, supply chain disruptions, and transportation barriers led to price inflation and essentials of cereals and reduced affordability for low-income people and migrant household people. Women and children faced nutritional deprivation, driving them to prolonged long-term periods. Suspension of midday meals and Anganwadi services further increased the food insecurity. Rural households reduced food quantity and quality due to the income loss, widening the gap between the availability and accessibility. However, the intervention helped reduce this impact, but the public distribution system, the PDS system, NFSA Act, the new National Food Security Act, and the Pradhan Mantri Garib Kalyan Anna Yojana, along with the integrated child developmental systems, supported vulnerable populations, whereas in a state like West Bengal, uninterrupted grain distribution was there, rationing through the coupons, police supported food decline. Police supported food delivery and the continuation of midday meals and child development. Services through home delivery.
You can conclude yours today.
In conclusion, I want to say that achieving zero hunger does not only require efficient distribution, reduced food wastage, and strong governance. Community participation, hunger in closely linked places led to poverty, inequality, and public health. But an integrated approach is essential to build a resilient and equitable food
Ananya, your
system
time is up. Thank you so
for a
much.
sustainable future. Thank you so much.
Ananya, how your study is contributing in scientific community? Can you comment on it?
So it is actually keeping everybody in touch, like how food security can be built in spaces. And like in COVID-19, we saw that there were lockdowns and people were not getting enough food in the starting. But due to some government programs, it was held in all states, and now it is available everywhere. Even some states like Odisha still continue to provide food grains to the house to migrant people.
Any question from Dr. Rajkumar?
No, continue. Next.
Thanks, Ananya.
Thank you.
So next candidate is Shivani Tripathi.
Hello.
Shivani, please share your poster.
Sir, am I audible, sir?
Yeah, you are. Kindly share your poster.
Okay. Sir, is it visible, sir?
Yeah, make it slideshow.
Yeah, I have already made, sir.
Proceed, please.
Can I start, sir?
Yeah, you can start, but now your poster is not visible. Kindly share again.
Okay.
No, it's visible. Please proceed.
Okay, sir. Yeah. Thank you, sir. Good evening to everyone. I'm Shivani Tripathi, SRF from CSIR Central Food Technological Research Institute, Mysuru. And today I'm going to present the poster on the topic entitled Coffee Husk as a Sustainable Source of Prebiotic Cello-Oligosaccharides for Gut Health. Every day, millions of us begin our morning with a cup of coffee. It's not just a beverage for us, but a daily ritual which we truly enjoy. But do you know, behind every cup of coffee, there is a hidden reality. Only 6% of coffee cherry ends up in the cup, while nearly 94% of it is discarded as a waste. So this has made coffee industry challenging and made us ask that this massive amount of coffee, can it be transformed into something valuable for human health? So for this, our background study has, after harvesting, coffee undergoes several processing steps. Firstly, the fresh coffee cherry is depulped into to obtain green coffee beans, which are then roasted and brewed to produce the coffee which we consume. However, during all this process, a large amount of byproduct is generated at several stages. Among them, coffee husk is the primary one. This coffee husk is generated approximately 0.18 million tons for every 1 million ton of coffee cherry. To understand its reutilization, its nutritional composition was essential. The coffee husk is lignocellulosic biomass, which is rich in cellulose around 45% and hemicellulose around 18%. Yet it is largely underutilized and often treated as waste.
Therefore, coffee husk make itself as an excellent candidate for valorization into functional compounds like cellulose-oligosaccharides. These cellulose-oligosaccharides are similar to other oligosaccharides such as xylo-oligosaccharide, inulin, fructo-oligosaccharide, which are commercially available. These are considered to be emerging second generation prebiotics with potential health benefits like prebiotic properties, antimicrobial benefits, and immunomodulatory effects.
You can conclude your poster.
Yes, sir. So, we had three different objectives. That was to develop efficient strategies for valorization and to evaluate their prebiotic potential and their contribution towards sustainable waste management approaches, sir. So we had two integrated pre-treat. We have an integrated pre-treatment methodology where we have used alkali pre-treatment along with enzymatic hydrolysis, followed by that. So we could achieve, and we have characterized 81.01% of cello-oligosaccharide mixture, which was cellobiose and cellotriose. These are mainly short-chain cello-oligosaccharides, which are known for its prebiotic potential.
Shivani, your time up. Thank you.
Yes.
So, which probiotic strain you have used for your study?
Sir, we have used Lactobacillus casei plantarum and Bifidobacterium longum, sir.
Okay, so how much is the maximum theoretical conversion by this probiotic?
Sir, conversion in terms of value we have not got. We have only checked the effect of this probiotic strains over gut microbial strains, sir. Like our supplementation of cello-oligosaccharides to probiotic strains.
Shivani, is there any negative effect on the gut microbes?
Sir, that negative effect we have not evaluated for our study, sir. Only probiotic or gut modulating effects we have studied, sir, as of now.
Thank you.
Yes, sir. Yeah.
Shivani, which strains is modulating more in your experiment? Which strain?
Lactobacillus plantarum.
Lactobacillus plantarum. Okay.
Okay, thank you, Shivani. The next presenter is Harshita Singh. Harshita, are you there?
Yes, I'm there. I'm sharing my screen.
Sure.
So is my screen visible?
Yeah, it is. Please proceed.
So, good evening, everyone. I would like to begin by sincerely acknowledging my faculty mentor, Dr. Shruti Khanna Ahuja, and Amity Institute of Biotechnology for their constant guidance and support. Today, I'll be presenting my work on protein nanoemulsions as a drug delivery vehicle. So the aim of this study was to develop a urea-responsive enzyme-triggered drug delivery system using protein-based nanoemulsions. Now, protein nanoemulsions are highly attractive because they're biocompatible, biodegradable, and it's very easy to functionalize. In the system, we have used urease so as to trigger the drug release, so that the drug release can occur specifically in a urea-rich environment, such as urinary tract infections, tumors, and other pathological sites. Now, let me briefly walk you through the methodology which is shown here. So we have basically first prepared the dispersed phase using 18% weight by volume aqueous solution of BSA, which has served as the protein matrix. Then we have prepared the continuous phase consisting of hexadecane oil with span-80 as a surfactant. The surfactant stabilizes the droplets and prevents the coalescence. Now, both the phases were introduced into a microfluidic device. Due to controlled shear forces inside the microchannels, the protein solutions breaks into micro droplets. And then the use of the microfluidics was mainly to allow precise control over the droplet size and uniformity. The droplets were then collected and protein denaturation was induced, which led to the solidification and formation of the protein beads. Then we have optimized the flow rate ratio of the dispersed and continuous phase. By adjusting these parameters, we have achieved the mono dispersed protein beads, which were basically essential for the uniform drug loading and its release behavior. And further, the next phase of a study is supposed to focus on the enzyme immobilization and drug encapsulation. The goal is to incorporate urease and therapeutic agents with these protein beads so that the drug release can be.
Almost up. Please conclude.
So, in conclusion, we have successfully formulated stable protein nanoemulsion and demonstrated controlled bead formation using the microfluidic approach. Now, this platform shows a strong potential as a stimulus responsive targeted drug delivery system. Thank you.
My question is, what is the mechanism of this drug delivery?
So the mechanism is to basically release the drug in a specific urea-rich environments, such as urinary tract, tumors.
Harshita, have you tested any drugs, like any specific drug you're targeting, or?
So we have like not yet decided, but yes, we'll be.
It's a review, literature review, or it's your own research?
Sir, it's a literature review.
Okay, thank you.
Thank you, sir.
Any comments, Dr. Rajkumar?
No.
Okay, you may leave, Harshita.
Thank you, sir.
So next is Priya Gupta.
Anju Devi. Ishani Mitra. Nikita.
Yes, sir. Am I audible?
Yeah. Please share your poster.
1 second. Is this okay, sir?
Yes.
Hello.
Okay, kindly put in presentation mode.
Just give me a second, sir. Is this okay?
Okay, you can start.
So, good afternoon, everyone. I am Sai Nikita from Department of Biotechnology from Chaitanya Bharati Institute of Technology, Hyderabad. So, our project is titled as Enhancement of Nutraceutical Properties of Pleurotus ostreatus through submerged fermentation with lactobacillus. So, Pleurotus ostreatus, commonly is known as oyster mushroom, is nutritionally rich and edible fungus, which is containing polysaccharides, flavonoids, and it also contains some essential amino acids. These compounds are known for antioxidant and immunomodulatory properties. However, many of these bioactive components are present in bound form, reducing their bioavailability. So our objective was to enhance its nutraceutical properties using lactic acid bacterial fermentation. Here you can observe in methodology, we took powdered mushroom. We took fresh oyster mushrooms, which were cleaned and powdered. Next, lactobacillus was isolated using a commercial probiotic source, that is Porlac. We prepared a nutrient broth and inoculated it with isolated lactobacillus culture under
sterile conditions. So here we are using submerged fermentation, in which we are introducing mushroom powder into the broth containing bacterial culture. Controlled and inoculated samples were maintained to compare the growth and fermentation effects. Fermentation process leads to microbial biotransformation, where complex phenolic compounds are broken down into more bioavailable forms. Next part will be continued by my teammate Pranit.
So good afternoon, everyone. My name is Pattabhirama Pranit Chaturvedula. So I'm here to explain about the expected results, like the before and after fermentation, the properties like the antioxidant properties and also the biotic compounds. Generally, the nutraceutical properties after fermentation is expected to increase. And as you can see in the graph, after the HPLC chromatographic techniques, the properties of the mushroom are being increased. And before the fermentation, the mushroom generally contains the proteins, the dietary fibers, the vitamins, and antioxidant. And after the fermentation with the lactobacillus, which is generally regarded as a safe bacterium, so it helps to increase the bioactive
Kindly
compound.
conclude, please.
Yes, sir. After fermentation, the properties like the phenolic contents and the antioxidant properties, which are useful for the persons who have digestive disorders, and also they can be cured with they get rid of the some properties like disorders.
So my question is, what kind of phenolic contents you have measured in your control and post fermentation?
So actually, sir, we are still done with the methodology. We did not test for the phenolics and antioxidants, but we are going to test, sir, with Folin reagent method.
Yes.
Or it will help you to characterize what kind of protein are present. I'm just asking because the phenolic content you are showing here.
Yes, sir. Can you repeat the question? I did not hear.
What kind of phenolic content were increased when you perform the submerged fermentation?
They are related to the gut health, sir, those phenolics.
Okay, just I will give you some hint. Whatever the fermentation broth you got it, just scan the UV spectrum. You will find so many peak and just find out what are the components you got it from the literature.
Okay, sir. Actually, we are at the bottom of our project. We are expecting our results in next month, sir, because the fermentation process is going on.
A quick question from methodology. What's the ratio of this mushroom and that sporelec you are mixing? What's the ratio?
It's basically 1:1, sir.
Okay. Okay. Thank you, Nikita.
Yeah, thank you, sir.
Thank you, sir.
Next is Riya Agrawal. He's not there. Amit.
Yes, sir. I am presenting.
Amit, please share your poster.
Sir, is my screen visible?
Kindly make it slideshow. No.
Sir.
Okay.
I think the screen is visible, sir.
Yeah, you can proceed.
Sir. Namaskar, sir. My name is Amit Vikram Pandey. I'm from MIT Law School, Noida, and I'm currently pursuing my BB LLB honors, and I am in my final year, 10th semester, with IPR, Intellectual Property Rights, as my specialization subject. The main reason for writing this paper and presenting it in this biotechnology conference is because biotechnology also stands under the ambit of IPR laws, intellectual property laws, gene editing, genome editing, and certain other biodiversity act and biotechnology laws come under the purview of intellectual property rights. The paper is CS. From lab to law and from lab to law, regulating CRISPR enabled green biotechnologies for sustainable development. What CRISPR stands for is clustered regularly interspaced palindromic repeats.
It's a time-bound presentation. Time-bound presentation. You no need to explain everything. Just
Certainly. CSI editing comes under the preview of IPR, and it is important because genome editing because of this, India's agricultural units and biotechnology has excelled a lot. But in the same space, it gives us concern about biosafety regulations. Precautionary principle is in our environment, so we need to keep that in our minds as well. If a gene is edited, it goes unethical, and if it is unethical, then it will be dangerous for the society. Sustainable development in Indian context is sustainable development occupies a central position in Indian constitution, jurisprudence and policy framework. The integration of environmental protection with ecological economic development has been repeatedly endorsed in the Indian courts and essential is to governance principle. CSIRP and patent laws has forwarded it. If we are editing a gene, it can be patentable. So we need to be very critical about giving what will be patented and what will not be patented. Because if we patent a gene of a seed of certain organism that is for the benefit of the people, then that will go against the laws. Then that will go against society. And sorry.
Kindly conclude your poster.
Certainly. Conclusion: the CRISPR enabled green editing represents a critical juncture where scientific innovation intersects with legal governance and sustainable development. India's constitutional values, environmental jurisprudence, and public interest-oriented patent design provide a strong foundation for regulating these technologies responsibly. By embracing adaptive, transparent, and science-informed legal frameworks, India can ensure that CRISPR innovations contribute meaningfully to the sustainable development while reinforcing the role as a global leader in ethical biotechnology governance.
Thank you, Amit. Your time's up.
Thank you so much.
Dr. Rajkumar, any questions from you? My question is that if somebody is doing genetic engineering using CRISPR, so how can they patent their strain?
Patent as in?
Patent as a strain, using strain manipulation using CRISPR.
सर नोवार्टिस एजी का एक केस था सर. नोवार्टिस एजी का अगर हम रेफरेंस लें इसके अंदर, उसके अंदर उन्होंने एक कैंसर की ड्रग थी ग्लेवेक करके, उसको पेटेंट करवाया, उनका पेटेंट था. उसके बाद जब उसका पेटेंट रिन्यू होना था, उन्होंने हल्का सा उसका स्ट्रिंग चेंज किया, मॉलिक्यूल उसका मॉडिफाई किया, फिर से पेटेंट कराने गए, वो पेटेंट नहीं हुआ क्योंकि कैंसर एक ऐसी बीमारी और उसके ऊपर वो 1.5 लाख पर मंथ, 1.5 लाख का उसका पर मंथ का खर्चा आता था, जिसके लिए कंटेंट किया गया कि ये सोसाइटी वेलफेयर के लिए नहीं है. पेटेंट इज फॉर सोसाइटल वेलफेयर एंड सस्टेनेबल डेवलपमेंट ऑफ द अर्थ. इंडियन पेटेंट काम करता है दुनिया अपने सिटीजन्स को हेल्प करने के लिए. तो इसका पेटेंट रिवोक किया गया ना, इट वाज नॉट गिवन अ पेटेंट, और उसका पेटेंट सर कंपल्सरी लाइसेंस भी किया गया था. तो पर्टिकुलर स्ट्रिंग को आप चेंज करके पेटेंट नहीं करा सकते, लेकिन पर्टिकुलर सपोज आपने इनोवेट किया है वो पेटेंट होगा. लेकिन इफ इट कम्स अंडर द एम्बिट ऑफ सोसाइटल वेलफेयर, अगर सोसाइटल वेलफेयर नहीं होगा तो वो कंपल्सरी लाइसेंस कर दिया जाएगा एज पर लॉ. ट्रिप्स एग्रीमेंट.
थैंक्स अमित.
थैंक यू सो मच सर.
Priyanshi. Okay. Swarnim, Rakhi.
Hello.
Rakhi,
Am
please
I
share your
audible?
poster. Yeah, you are audible.
Thank you, sir. Select entire screen.
My screen is visible, sir? Hello?
Not visible.
Visible or not?
Yeah,
Now.
please make it in a presentation mode.
Oh, is it okay, sir?
Yeah, it's fine. You may proceed.
Okay, good evening, sir. I am Rakhi Kapoor from Microbiology Department of Guru Nanak Dev University. My topic is fermentation of beetroot juice using lactic acid bacteria and utilization of wheat waste for biogas production. So why we choose beetroot for probiotic drink? Because most probiotic beverages available in market are milk based. However, these products are not suitable for lactose intolerant individuals. Therefore, there is a need for non-dairy probiotic alternatives. So beetroot is rich in antioxidants, having bioactive properties and a promising non-dairy fermentable substrate and potential substrate for integrative food and bioenergy production. Objectives of these studies were development of plant-based probiotic beetroot beverage, evaluation of lactic acid production efficiency, and validation of wheat waste for biogas production. Methodology of this study was beetroot juice were extracted uh, and fermented with the help of lactic acid bacteria. For the fermentation, 5% lactic acid bacteria are inoculated and fermentation were carried out at 35 degrees Celsius temperature. After fermentation, after the fermentation, pH and acidification of the juice were acidification and viable cell count were examined, and sugar utilization and lactic acid production were estimated. Sensory evaluation was also performed, and the residual biomass was further used for the biogas production. The results of the studies are from three strains of lactobacillus lactic acid bacteria. L18 exhibit highest lactic acid production, which were 1.72% at 2% xylose. Significant acidification achieved from pH reduction from 5.23 to 3.88. Viable lab maintained at 10 to the power 5 CFU per ml. Provital, which name were given to the probiotic drink, showed acceptable sensory performance. Enzyme assisted digestion enhanced biogas yield, which is 145 ml, which is much higher than the control, which was 85 ml.
Okay, kindly conclude your poster. Rakhi, your times is almost up. Kindly conclude.
So this study demonstrated probiotic beverage and sustainable utilization of the Hello?
Yes, Rakhi. Kindly conclude your slide.
So this study highlights a sustainable and value-based strategy for functional food development and waste management. Thank you so much.
My question is how much is the sugar content in the beetroot and how much it is converted into lactic acid by lactic acid fermenting bacteria?
Sir, can you please repeat?
How much sugar content in the beetroot and how much sugar is converted by the lactic acid fermenting bacteria?
Okay. So here is a table. The lactic acid production were carried out in different concentration from 1% xylose, 1% 1% xylose, 2%, 3 different. This is the these are the results.
Okay, thank you, Rakhi.
1.72% result lactic acid produces with the L18 strain.
Okay, thank you, Rakhi. We got your point. You may leave.
Thank you, sir.
Rashi Chauhan.
Yes, sir. I'll share my screen now.
Yeah, please.
Is it visible? My poster is
Make,
visible.
make, make it slideshow. Yeah. It's not in
Can
the
I start now?
Yeah, you can proceed.
Good evening, respected panelists and all the participants. My poster is titled Valorization of Lignosulfonate Waste into High Value Enzyme Nanocarriers for a Circular Bioeconomy. Lignosulfonate is a major industrial waste from the sulfide pulping process and is usually discarded or burned, causing resource inefficiency. In this study, we aim to convert this low value lignin waste into biodegradable nanocarriers for enzyme immobilization using a sustainable waste to wealth approach. First, sodium lignosulfonate was purified and dissolved in water for 24 hours. For this complete dissolution, a CTAP solution was then prepared, and due to electrostatic interaction between negatively charged lignosulfonate and positively charged CTAPs, self-assembled lignin nanoparticles were formed. Then dropwise addition of molecular water helped control the nanoparticle formation and stabilize uniform colloidal nanospheres. Next, a multi-enzyme cocktail was produced and extracted and immobilized onto these lignin nanocarriers through mild absorption at room temperature. The enzyme-loaded nanocarriers were recovered by centrifugation, where the formation of a visible pellet formation confirmed a successful synthesis. And for characterization, UV visible spectroscopy indicated nanoparticle formation and enzyme loading through absorbance changes. FTIR analysis showed characteristic lignin peaks, and after immobilization, noticeable peak shift confirmed enzyme binding onto the nanocarriers. DLS results revealed that the initial lignin nanoparticle exhibited a broad size distribution with peaks around 241 nanometer and 1232 nanometer, indicating polydispersity. But after enzyme immobilization, a more uniform particle size around 780 nanometer was observed, confirming successful loading and stabilization of the enzyme nanocarriers. So overall, the study demonstrates a green and cost-effective strategy to transform lignin waste into functional biodegradable enzyme nanocarriers. So this is it from my side.
Okay, Rashi. So how much nanoparticle you have produced from lignin? What is the yield of that nanoparticle? And
so we aimed for 1 mg per ml at the end of like after centrifugation. We aim to achieve the concentration of 1 mg per ml.
In terms of yield, means how much lignosulfonate you have taken and how much nanoparticle you have.
So we took 0.5 grams of solid NALs and lignosulfonate, and after adding the molecular water dropwise, we got the concentration of 10 ml. And after centrifugation, we converted it into 1 ml.
No, your input is 0.5 gram and your output is in ml.
Yes, sir.
Okay.
We added.
Thank you, Rashi. Dr. Rajkumar, any question? Rashi, you may leave.
Thank you, sir.
Priyanka Sharma.
Yes, sir, I'm here.
Please share your poster.
Yes, sir.
Priyanka, share your poster.
Sir, is it visible?
Zoom it, please.
Can you make it zoom?
Yes, sir.
Yeah. Okay, fine. You may
Yes, sir.
proceed.
Thank you. Good evening, respected judges. I am Priyanka Sharma from the Department of Bioengineering, Techno India University, Tripura. Today, I am presenting my poster on edible insects as a significant source of food and nutraceuticals. So, starting from the introduction, due to rapid population growth, traditional food systems are becoming unsustainable. Livestock farming uses a lot of land, water, then energy, and also it causes a lot of pollution. So, therefore, we need alternative and sustainable food resources. Then, for this reason, edible insects are a good solution. They are rich in protein and healthy fats, then vitamins and minerals like iron and zinc. They also contain bioactive compounds which help the health to improve and can be used as nutraceuticals. Then, moving towards the methodology section of the poster, edible insects can be processed by simple methods like drying, roasting, grinding into powders, as well as modern technologies. As these methods, through these methods, we can easily use this use this in food products. Mm, in terms of application, they can be used as human food, health supplements, and also animal feed. Mm, and insect farming also need less land and water and produces fewer greenhouse gases. It also helps us with economically as it can create a lot of income and job opportunities to people. So, in conclusion, edible insects offer a sustainable, nutritious, and eco-friendly solution to future uh, food challenges. With proper awareness and support,
Priyanka, kindly
they can
conclude
help.
your poster. Kindly conclude.
Yes, sir. This is my conclusion. Edible insects offers a sustainable, nutritious, and eco-friendly solution to future food challenges. With proper awareness and support, they can help ensure food security. Thank you, everyone, for your attention.
You are using edible insect. Can you tell us the composition of edible insects?
Sir, can you please repeat the question?
Can you tell us the composition of edible insect?
The composition of edible insects. Sir, edible insects have a lot of nutritious components such as protein, fats, vitamins, minerals like iron, zinc, fiber.
Can you tell us the composition in percentage? Which one is higher? So we can use edible insect for the purpose of food and other nutraceutical purpose.
Sir, sir, I'm not hearing the question properly.
No, sir is asking like any insect, what is the composition of like carbohydrate, sugar, protein, which is most like protein is more, so we can use it as a protein source.
Sir, the protein composition is more.
How much in percentage?
More is just a relative term. No? So if I'm saying 25% protein is present in the pulses, so just like same in edible insect.
Sir, this can be different in different insects.
Okay, thank you, Priyanka.
Priyanka, I have one question. Have any example that is available in market?
So sorry?
Have any examples of this type of food that is available in market?
Sir, there are like crickets, worms, which are edible for humans.
That is available in Indian market?
No, sir, not
Okay.
really.
Not have any product that is available in Indian market, no? Till now.
No, sir.
Priyanka, I must say, just because your rationale is key, we are increasing our population, we can't lose the diversity also. Got
Okay,
it?
sir.
Oh, yes.
Okay, sir.
Please
Thank
take
you.
this in mind. Okay. Thank you, Priyanka.
So can I leave?
Vineet. No. Deepti.
Priyanka, you can leave.
Okay, sir. Thank you.
Thank you so much, Dr. Rajkumar. We'll facilitate you.
Thank you. Thank you, Dr.
And we'll
Krishna.
meet in person.
Thank you. Thank you all.
Thank you, Dr. Rajkumar, for your time.
Thank you.
It's now my privilege to invite Dr. Ajay Chauhan and Dr. Yashika Reheja to the stage to honor and facilitate our respected chair for their valuable presence and guidance. Dr. Krishna Gautam, sir.
Participants, we are concluding with the meeting. Thank you for joining.